The second session now, we're starting to delve into some of the actual biomarkers. And I'm starting with the Sympatho Adrenal Medullary system, a little bit of that system and the things you can measure and different ways of measuring these markers. So let's see. Need to get everything set here. All right. So we'll start with this table, which shows some of the behavioral and physical or physiological, I would say, adaptations to an acute stressor. So I'm going to focus over here on this slide. So there are changes in blood flow that direct nutrients and oxygen, nutrients being mainly glucose, but also fatty acids, to the brain and other body sites, such as skeletal muscle, increased blood pressure and heart rate, increased respiratory rate, increased gluconeogenesis, which is the de novo synthesis of glucose in the liver, and lipolysis, the breakdown of fat stores by the liver. So both of these are geared toward providing, rapidly providing increased energy, inhibition of growth reproduction, et cetera, et cetera. So most of this stuff here, these rapid responses to an acute stressor, are mediated by activation of the Sympatho adrenal medullary system. And so that's why, if you're interested in acute stress, this is a system that you should know about and definitely want to take a look at. Tomorrow, when you hear from Katie Dixon-Gordon, you'll see alternate ways of studying that system. So here we're looking at chemical or hormonal biomarkers of this system and its responses. So this is a review of the sympathetic nervous system. So here are, this is a chain of ganglia that run near the spinal column here, preganglionic neurons here at the thoracic levels, T1 down to T12 and below, and innervating these ganglia. And then the sympathetic ganglia innervate, so these are nerve fibers here, postganglionic, sympathetic nerve fibers that innervate the pupils. So pupils are dilated to let in more light. Saliva, which is important to begin digestive process, is inhibited. The bronchi are the smooth muscle that determines the amount of opening of the bronchi are relaxed. The smooth muscle is inhibited. This allows more air to come in. The heart rate is accelerated, of course, to speed blood flow. Peristaltic activity, smooth muscle in the gastrointestinal system, is inhibited. So this is part of the general inhibition of digestion. I mentioned glucose production and release by the liver. And importantly, down here, an increase in release from the adrenal glands over here of epinephrine and norepinephrine, or adrenaline and noradrenaline. And if you heard the term, you probably did at some point in school or in the popular press, the fight or flight response. This is what we're talking about, these rapid physiological reactions to an acute stressful event. So we're going to be talking here in this second session about one part of the adrenal gland, which is the adrenal medulla. That's the interior part of the adrenal gland. And this shows a nerve pathway that goes directly to, that innervates the cells. They're called chromaffin cells, for those of you who might not be familiar with that. Chromaffin cells are the cells that synthesize epinephrine and norepinephrine. They're sort of like neurons in their biochemistry, except that they don't have dendrites and axons and things like that. They're secretory cells. So instead of secreting these into synapses as neurotransmitters, these substances are secreted into the bloodstream as hormones. The other part, of course, that we're going to be talking about at length this afternoon is the adrenal cortex. Cortex means bark, the outer part of the adrenal gland, and that's responsible for secreting glucocorticoids like cortisol, mineralocorticoids like aldosterone that regulate salt retention. And we'll be talking about all of this stuff later. So for now, we're interested in this. Because there's a direct nerve connection here, this is what allows this part of the adrenal gland to respond so quickly to stress. This takes longer because there's a cascade of hormones that all need to be released and to act on their respective targets. So this system is very important for the short-term stress response. So SAM, Sympatho, adrenal medullary system, rapid onset and offset. So that squirt of catecholamines that's released into the blood, it peaks very quickly, and then when the stressor is over, the levels go down also very quickly due to metabolism and clearance of those hormones. The HPA system, as I said, we're going to be talking about a lot this afternoon. Slower onset, but more prolonged response. And these, in fact, were evolved to coordinate with each other in their effects on cardiovascular system, their effects on energy metabolism, like glucose and fat metabolism, and so forth. So biomarkers of Sympatho adrenal medullary system that we're going to be talking about now. So since epinephrine and norepinephrine, epinephrine, by the way, is the major one. There's considerably more. Norepinephrine is much more important as a neurotransmitter in the brain. There's only a few clusters of cells that in the brain that neurons that use epinephrine as a transmitter. So this one's much more important in the brain, but in the from the adrenal gland, much more of this is released than this, but nevertheless, both are. And this norepinephrine is also a transmitter at, if I go back here for a minute, that is, norepinephrine is the transmitter that's released by all those nerve endings. Oh, actually, except for the salivary glands, that's the one exception. But for the most, you don't have to worry about that. For the most part, norepinephrine is the mediator of sympathetic nervous system activation on the target organs. So we can measure these in plasma. Also, if you want to know about a longer acting stressor, something that might persist in its activation of the adrenal medulla and the sympathetic nervous system, you can collect 24-hour or less, but often it's 24-hour urine from your participants, and you can measure the excretion either of these substances unmetabolized, but more typically metabolites, since they're subject to substantial metabolism before they're excreted through the kidney. And then, as an alternative to either collecting blood or collecting urine, you can also measure a marker of sympathetic nervous system activity using an enzyme that is secreted in saliva called alpha amylase. And so we'll be talking about that, since that is widely used in many stress studies as an alternative to measuring these. So what if you want to measure plasma epinephrine and norepinephrine? It might be that none of you will ever do this, but it's good to know about it as an alternative. It's more likely to be done in a medical setting, maybe a hospital setting or a clinic. One of the reasons is because the blood sampling needs to be done very rapidly. So you'd be likely to have your participants who are going to be stressed with an indwelling catheter. So you'd have someone, a nurse or a doctor or someone, put in a catheter ahead of time so that you can draw blood repeatedly without having to stick the participant with a needle over and over again. Anyway, so it is appropriate. This is appropriate for acute stress studies, but you need to get your blood right away, so you likely have an indwelling catheter. As I mentioned, the epinephrine is that you're measuring in blood is mostly from the adrenal medulla, whereas norepinephrine is mostly from the sympathetic nervous system spillover. So what do we mean by that? So those nerve endings that I mentioned in the previous cartoon, where you've got these sympathetic neurons that have, axon terminals near, let's say, smooth muscle. It could be vascular smooth muscle. It could be cardiac muscle, if you're talking about the innervation of the heart. Some of the neurotransmitter that's released, this norepinephrine, some of it is taken up, back up into the nerve endings, but a lot of it escapes and it finds its way into the bloodstream. So this amount of neurotransmitter that was released that escapes from reuptake is what we call spillover, and can be used as an indicator of how much norepinephrine was being released at those nerve endings. And there's several different ways, if you're interested in the technology, either to set it up in your lab or if you're working with a collaborator. Routinely, these substances are measured using HPLC. This is an HPLC setup here, or by ELISA, or by radioimmunoassay. Those would be the big three ways of measuring these substances. Now, since we're talking about a lab setting, because again, you might have your participants catheterized, in which case it's definitely in a lab. But even if not, and you want to evoke an acute stress response and then measure whatever markers you want to measure, there's several different kinds of tests that are widely used for stress induction in the lab. So one that we've actually used here, I collaborate with a faculty member over in the College of Nursing here at UMass, Karen Kalmakis. And in one study we did a few years ago with nursing students, one of the things we subjected them to was the cold pressor test. This goes way back. And the idea of the test is that it's a physical pain. So the participant puts their hand into ice water. Either it could be done for 1 minute or until the pain is unbearable. They're told to keep the hand in there until you can't stand it anymore. And you can show that you readily evoke cardiovascular responses. That's why it's called, so pressor refers to blood pressure. So this, so cold pressor means that it was first identified as being a test, a stressor that elevates blood pressure, Sympatho adrenal medullary activity, and also HPA cortisol responses. And of course, you can also measure the pain intensity, see if that correlates with any of the biomarkers that you're measuring. If you're interested in a psychosocial stressor, which is done more frequently, then probably the most common one was actually developed by Clemens Kirschbaum, who I mentioned earlier. And before he moved to Dresden, where he is now, he was at the University of Trier in Germany. And his group developed a pretty effective laboratory stress test that they called, since it involves other people, social setting, they called it the Trier, it came to be known as the Trier Social Stress Test, or TSST. And there are two stressors in this task. One is public speaking. Typically, there are a couple different ways you can do this. There's always more than one way to skin a cat. But one of the common ways is to instruct your participants that they have to interview for a job. This is a really important job. They really need this job. You give them several minutes to write up a presentation about why they're the most qualified person to be hired for this job. And then before they give the presentation, you take their notes away. So now they have to do it from memory without the benefit of their notes. And you'll have typically several white-coated, at least the ones that I've seen, white-coated investigators, research assistants, whatever, sitting behind a table. You're at like a podium and you have to give this presentation to them, and they're looking at you stony-faced, not giving you the normal, head nodding and positive social feedback. It's meant to be as intimidating as possible, okay? And if you stop, if you can't think of anything more to say, you're instructed to keep going. So they keep the pressure on. And that goes for several minutes, like 5 minutes here in this exemplar here. And then you have a mental arithmetic problem. So the common one is you're given the number 1022, and you have to, in your head, subtract 13. So you subtract 13 from this number, and then you subtract 13 from the resulting number, and you keep going back and back. And if you make a mistake, they frown. Well, I don't know if they frown, but they tell you start over. And so again, it's meant to be, in a way, it's meant to be intimidating and scary. There's a number of different measures that are often taken during the test. So here's a waiting period here. We'll talk about this. This is important, this waiting period here. So people come into the lab. In this particular example, this is actually a paper by Melissa Burkett, where she lays out a paradigm for conducting the Trier. So she has a fairly long waiting period here. We can talk about why that is. Then there's the pre-stress period, when they will typically do some psychological measures, any number of things. And then you do, let's see, so then the AS is the anticipatory stress period. This is where you're preparing a speech, 5-minute speech, math period, so 5 minutes of doing that, and then recovery. And so for this paradigm that's being suggested here, she has saliva samples collected here, arrival in the lab, here at the end of the waiting period, at the end of the, so this is like, this is a baseline, from coming to the lab. This is your baseline before the testing starts. Alternatively, you could put it over here. There's variation here. And then post-stress, and then usually at least a couple of recovery samples to look at how rapidly your salivary cortisol is going down. You can measure heart rate. You can measure plasma, epinephrine, and norepinephrine if the participants have been catheterized. Of course, you can have them fitted for heart rate and blood pressure. You can also measure alpha amylase as an indicator of sympathetic nervous system activity. And then if you're collecting saliva, as shown here, you can get cortisol or DHEA, as we talked about a few minutes earlier, in along with DHEA in your saliva samples. I'm going to skip that. So here's, this is just some example data of cardiovascular and hormonal responses to the Trier. So here's the design of the study. It was 44 women. And in this particular study, it wasn't important to us necessarily, but the authors were interested in whether having differing degrees of fitness affected your stress responses in this test. They were catheterized, and plasma hormones, so epinephrine, norepinephrine, and cortisol were measured before, during, and after that, as well as heart rate and blood pressure. So here's heart rate. So here's the low and high fitness groups. There weren't big differences between the groups, as you can see, and it's not that important for us. But you can see here's the test itself. Nice heart rate response on average. Here's plasma cortisol, a very nice plasma cortisol response. And note that in this particular study, they took a lot of blood samples because it was easy with the catheter being in there. And so they really got very detailed data on recovery from the stress response, the cortisol stress response. Here's the plasma adrenaline or epinephrine. Not much there, despite the heart rate increase. So this shows you that right off the bat, different measures of reactivity of the system can vary, can differ. There was a much better response of norepinephrine. So the sympathetic nervous system did respond, but the adrenal medulla, this part of the system, didn't do much. So if you want to measure urinary epinephrine and norepinephrine, that's appropriate for either acute or chronic stress. The reason why I say it could be used with acute stress is you might have a situation where you're expecting, fairly prolonged responses to stress. And so in that case, you might want to see how much epinephrine and norepinephrine are being secreted over time. And in this case, excreted in addition to secreted. You'd have 12 or 24-hour urine collection. There's a jug, a funnel, so you have to pee here and collect it, and then return it to the lab. And the hormone concentrations can be affected by the production rate of urine. So if you're just taking the urine and you're measuring whether it's cortisol or whether in this case it's epinephrine and norepinephrine, if the individual is producing a lot of urine over that 12 or 24-hour period, it's going to tend to dilute whatever amount of the hormone was excreted, and this will influence the absolute concentration that you measure. And so you need to control for that. And so you can control for urine production rate by measuring creatinine, which is a substance that it comes from muscle metabolism and is excreted into the urine more or less proportion to the rate of urine formation. So there are simple tests that you can buy, lab tests for measuring creatinine content of your sample. And you just correct for the amount of creatinine. So here was just one study that I happened to pick out. It was long distance bus drivers in the Netherlands. And the researchers collected urine samples for 3 days during long distance trips that were followed by, each trip was followed by 2 days off. They analyzed for epinephrine and norepinephrine, and again, corrected for, well, they're corrected for urine, but were expressed as excretion rate. So the data look kind of odd, but and there you can see big error bars. So there's lots of variability between different drivers. But here we see the day one. The gray bars are working days. So a big spike. This is the epinephrine or adrenaline. This is the noradrenal or norepinephrine. See a good spike in the over time. This is they collected at 3 or 4 hour intervals, the urine samples. So a big spike in epinephrine. Day 2, a little bit noisy, but again a spike, and then another spike on day 3. Day off, lower level, and day 2 off, again lower level compared to these. So here was the norepinephrine, and again you see a somewhat similar pattern, except that with changes over time, except that there really wasn't much of any difference from the working days, more stressful days compared to the off days. And so the norepinephrine excretion was relatively unaffected compared to the epinephrine excretion in this particular study. So again, just some example data, show you one way that it was used. Now, it's much more common nowadays, if you are interested in looking at sympathetic nervous system responses to stress, it's much more common to do this using saliva. You don't have blood samples, you don't have to catheterize your folks that you're studying. All you need to do is collect saliva samples. And of course, the beauty of it is, as we'll see, is that you can use those same saliva samples to measure cortisol. There still are some tricks involved, which we'll talk about when we look at cortisol responses to the Trier. But the point is you can get both. So here we're just going to focus on the sympathetic measure, which is salivary alpha amylase. So let's look at the saliva glands. So this is also relevant for cortisol, of course. So saliva production is from three glands here. And under resting conditions, 65%, or roughly 2/3, is from the submandibular gland down here, under your jaw, submandibular. Sublingual will be under your tongue, and the parotid is the big one up here. Under stimulated, that is stressful conditions, over 50% of saliva production now is from the parotid gland. So this is the one that's particularly responsive to stimulation. So what is alpha amylase? So this is an enzyme. So it's a protein, it's an enzyme, and it is the first enzyme that starts to break down starch. So right away, as soon as you eat a meal that has any carbohydrate or complex carbohydrate in it, it already starts to get broken down before it even hits your stomach or your small intestine. It's secreted by alpha amylase is secreted by a type of cell within the salivary glands called the acinar cells. And as you might expect from this information, the large majority of the alpha amylase is produced by the parotid gland over here. So it's important, if you're going to use this kind of biomarker or you're thinking about it, it's important to be aware of the fact that the salivary glands receive innervation not only from the sympathetic nervous system, which is what you may be interested in here, but also from the parasympathetic system. So these are, of course, the two arms or the two branches of the autonomic nervous system. So here's the salivary gland here, here's the parasympathetic innervation here, and here's the sympathetic innervation. So the parasympathetic innervation, as I say, is mainly effective or mainly responsible for controlling the rate of saliva production and release. Not entirely, but that's its main function. Whereas the main function of the sympathetic innervation is not only the rate of secretion, but, it is the rate of secretion of salivary proteins, such as alpha amylase. So that's why we can, to a reasonable extent, with limitations as I'll show, But to a reasonable extent, we can argue that this is a marker of activation of the sympathetic system. So what's the actual evidence that this is from the sympathetic system other than my just saying so? So this is an animal study. I'm not going to give you very many animal studies today, but occasionally when it's relevant. So in this case, what was nice was that the researchers could subject rats either to stimulation of sympathetic fibers innervating the salivary glands alone, or parasympathetic fibers alone, or both. And this is compared to baseline. 0 is baseline here. And you can see that actually sympathetic alone didn't do much, and parasympathetic alone did a little bit, but the two together. So you actually need an interaction of the two systems to get a big increase in amylase release. So that's the first thing to be aware of. So it is mainly from the sympathetic, but it requires additional interaction with the parasympathetic system. So what about the response of this marker to stress? So here we have our Trier stress test shown in the shaded area here. So here's a baseline sample, the amount of alpha amylase in the saliva sample. Here's post-stress, and here's recovery at 20 minutes and at 40 minutes. So this is the alpha amylase. And can we show any relationship between alpha amylase and our traditional markers of Sympatho adrenal medullary activity, namely norepinephrine and epinephrine? So in this particular study, they collected blood samples for measuring epinephrine over here and norepinephrine over here. They both responded in terms of increase in blood level. The question is there a relationship between the amylase response and the response either of norepinephrine or epinephrine? And what this shows here is that there's a modest but significant correlation between amylase and norepinephrine. There was no significant correlation with epinephrine. And that's okay, because again, keep in mind that the norepinephrine is mostly from the sympathetic nerve endings, spillover, remember? Whereas the epinephrine is mostly from the adrenal medulla itself. So this is by no means perfect. We'd love this to be higher, but it is significantly correlated, and it's correlated with the right marker, the marker that we would prefer, namely the one of sympathetic activation. So how do we do this? How do we measure amylase? We'll talk about collecting saliva. Actually, we're going to talk about collecting saliva this afternoon when we talk about cortisol. But you collect your saliva samples, and then you have, you're usually doing this by ELISA, or let's say enzyme immunoassay is the more generic term. And Cellometrics, we're talking about them more this afternoon as well. Any saliva biomarker, the first place that you should think of looking for measuring almost anything in your samples is Cellometrics. They're a very good company. They do a lot of research themselves. They have a huge number of kits that are available at, I would say, fairly reasonable prices for measuring a whole bunch of things. They can give you very good advice on study design, on how to collect saliva samples properly, and so forth. So it's a very, very excellent resource. There are some other companies too, but Cellometrics is the first place to look. Anyway, so we already saw that it's not a pure measures of sympathetic because parasympathetic nervous system is also important. In the bibliography that I provided for you, there are a couple reviews. They're not super recent, but they're quite extensive from the time of publication about using alpha amylase as a biomarker. There's another paper, a little bit more recent, reviewing application of amylase to psychopathological disorders, especially anxiety-related disorders. And then also a paper, not the most recent, but still quite good, that talks about the caveats and limitations of this approach from Bosch et al. So those are, again, if you're interested, those are references that are available in my folder on the OneDrive. So to summarize, as I said, we were going to go through this a lot more quickly than the first talk. To summarize, if this is a system you're interested in looking at biochemically, again, you're going to get other approaches tomorrow morning. I think it's in the morning. Anyway, you're going to get other approaches tomorrow from Katie Dixon-Gordon. But if you want to do this biochemically, such as with saliva samples, you can do that using alpha amylase. If you want to look at longer term responses, certainly if you want to look at chronic stress, you might want to look at urinary catecholamine measurements. And these are some of the ways that you're going to learn about tomorrow as alternatives. So that wraps that one up. So questions?
So I was seeing on your slides that norepinephrine and non-epinephrine, sorry, the adrenaline.
Norepinephrine and epinephrine?
Yes, thank you, sorry. They have different measures of, so they're not excreted at the same, not necessarily the same level, but quantities of it?
Right. Okay, so first of all, so the way we want to think about this is in a couple of aspects. So first of all, we want to think about where each one is coming from. So the both are secreted by the adrenal glands, the adrenal medulla, but with epinephrine predominating. Okay. So epinephrine more in the blood is almost entirely, or urine, is almost entirely from the adrenal medulla. So that's a relatively straightforward one. You're measuring epinephrine, you're looking at hormone that's secreted by the adrenal medulla, and very little of it is coming from other sources if you're measuring in the bloodstream. Norepinephrine is more complicated. A portion of it, a less than 50%, I would say, a portion of it is from the adrenal medulla. The majority of it is from sympathetic nerve endings. Norepinephrine that was released when the sympathetic nervous system was stimulated. It's nerve endings that are impinging on various organs, the heart, smooth muscle, vascular smooth muscle, that is smooth muscle around arterioles and things like that, digestive glands, bronchi, etc., all various organs. Some of that norepinephrine leaks out, gets into the blood, and you can measure it there. So the important point is twofold. First of all, the different sources. And second of all, that the adrenal medulla doesn't always respond in the same degree or to the same extent as the sympathetic nerve endings, the sympathetic ganglia. They're controlled by the same system, but the controllers, the controlling nerve fibers, are separate. And so this raises the possibility of somewhat differential responses. And so that's why you can more or less use one or the other or both, because they're giving you a different, they're a marker, a biomarker of two different things. The epinephrine, think of it as a biomarker of the response of the adrenal medulla, and norepinephrine mainly as a biomarker of the sympathetic nervous system activation independent of the adrenal medulla.
Thank you.
So,
And
yeah.
as a follow-up question, so when they place both of them on the chart, is it more of a standardized? Have they already standardized both of them as to, or is it more
Not
just
necessarily,
a proportion?
not necessarily, because they could be, but they're not so far different in the units of measure that you could put both of them on the same graph. So let me see. Let me look back at that tree. I'm just going to go back a couple of slides. Oh, I take that back. I take that back. I shouldn't have said that. I was way off. The epinephrine is much lower. I apologize. So the epinephrine is over here. So you can see this is the units of measure are both picograms per mil, but the epinephrine here, the range is 25 to 55, and the norepinephrine is way, way higher. You see a range of 100 to 500. So the reason that they were able to put them both together is because they had two axes. Sorry, that was definitely a misstatement on my part. But
Thank you for clarifying that.
Yeah, sure. Yeah, two different axes. Excuse me. Other questions?
I have a question, please. You had a graph where you showed that the combined was stronger than the one individually. So when you were looking at effects of the parasympathetic and the sympathetic, when they were together, it was stronger. So
that was a rat study.
Yeah, that one right here. So I'm wondering, what does that mean? Does that mean that it's a regulation effect?
Yeah. So so what will happen is that many stressors will actually tend, acutely anyway, will tend to inhibit parasympathetic activity, but it's not totally shut down. So what's probably happening here, probably the way to interpret this is that this is, what's the word I was looking for? Permissive. I think the way I would think about this is that this is permissive. You need to get a full-blown response, you need this going on to some extent, but the driver. So if this was higher, see, they purposely look at this is the nerve stimulation. So they were stimulating at 1 hertz, that is 1 electrical stimulus per second, whereas the parasympathetic fibers were being stimulated at 5 hertz, 5 stimuli per second. So they purposely made the sympathetic stimulation low so they could show the interaction. If this was at 5 hertz, you would probably see a much higher response over here. Maybe not as high as this, but you would see a higher response. So they purposely kept this low so that they could show the interaction.
Okay, thanks.
Any other questions? All right, so actually we got back on track. We're a few minutes early. So we'll be resuming, we'll be starting up the afternoon session at 1 o'clock sharp. So I hope to see you all there. So thanks for your attention this morning. Okay. So we're going to start with basics. Some of you may be quite familiar with this information already, others less so. And so I want to make sure that everybody's on as much as possible on a level playing field. So we're going to start up here in the hypothalamus. There are specialized secretory neurons within this brain area, the PVN, that secrete two peptides, the most important of which is corticotropin releasing hormone, or CRH. Sometimes it's also called corticotropin releasing factor, or CRF. So if you're familiar with that term, it's the same thing, CRH, CRF. And also arginine vasopressin, which is involved in the HPA axis, but it's better known for its effects on the kidneys, on renal function. Anyway, these two together, again, the main player being the CRH, travel down the stalk, the pituitary stalk here, to the anterior lobe of the pituitary, where there's a cluster of non-neuronal secretory cells that, when activated by CRH and AVP, secrete the next hormone in the cascade, which is adrenocorticotropic hormone, or ACTH. And the ACTH is secreted into the bloodstream, into the systemic circulation. And when it reaches the adrenal glands down here on top of the kidneys, the ACTH provokes the synthesis and release of glucocorticoid hormones, of which the one we'll be talking about mostly is cortisol, which is the main glucocorticoid in humans and in non-human primates and most other large animals, or corticosterone, which is the main glucocorticoid in rodents, birds, etc. And the reason that these are called glucocorticoids is because one of their functions, one of the earliest known functions, was on the disposition of glucose, or sugar, and hence the term glucocorticoids. And the other thing that the diagram shows is that once it's released, cortisol or corticosterone exerts a negative feedback action. So there are receptors. We're going to be talking about the receptors in a moment. The receptors for glucocorticoids here in the anterior pituitary and up here in several areas of the brain, which, when activated by glucocorticoids, tend to inhibit subsequent release of CRH. And therefore, they dampen the system. They prevent the stress response from continuing, assuming that it's an acute stressor, and allows for recovery of cortisol concentrations back to baseline. It's good to know a little something about the receptors, even if it's not something that you're directly studying. There are two subtypes of glucocorticoid receptors. There's one that's actually called the glucocorticoid receptor, or GR. The other is called the mineralocorticoid receptor. So mineralocorticoid reflects the action of hormones like aldosterone, which is also secreted by the adrenal cortex, and acts, I think I mentioned this morning, acts to promote sodium retention by the kidney. And so hence sodium, hence mineral, hence mineralocorticoid. Despite the fact that that's called mineralocorticoid receptor, it does bind and activates and is activated by glucocorticoids, at least cortisol anyway, not all glucocorticoids. So GR has a broad tissue expression. Most organs in the body express this receptor, which allows cortisol and other glucocorticoids to have very, very diverse effects throughout the organism's tissues, including the brain. One of the interesting things about it, the important things about GR, is that relatively speaking, it has a relatively low affinity for cortisol. And what this means is that at baseline cortisol levels, resting levels, when you're haven't been stressed, there's only a modest, minimal or modest occupancy of that receptor. Okay, so in other words, most of the receptor sites are not occupied, are not being activated by cortisol under non-stress conditions. And so this allows the receptor, the reason for that is it allows the receptor to be responsive to increasing levels when you're under stress. By contrast, the mineralocorticoid receptor has much more restricted tissue expression because aldosterone works in the kidney. Obviously, there's a lot of MR in the kidneys. In the brain, it's mostly restricted to the hippocampus, an area that's very, very sensitive to glucocorticoids. And this receptor, in contrast to GR, has a high affinity for cortisol. So receptor occupancy is high even at basal levels. So this receptor is not terribly stress sensitive. GR is much more important in that regard. So so we're going to focus our attention on GR. So when it's not binding cortisol or another glucocorticoid, the receptor, of course, the receptor is a protein, as almost all receptors are, whether for hormones or neurotransmitters or other signaling agents like cytokines or whatever. Almost all proteins. So this is a protein, and it's present, you can see, in the cytoplasm of the cell. And it's bound in the resting state. It's bound to several other proteins, including this one here, heat shock protein 90. 90 refers to the molecular weight of the protein. And what happens is, so this is meant to show the cortisol molecule or some other glucocorticoid, readily diffuses across the cell membrane, interacts, it binds to a site, a specific site on the receptor. These other proteins, heat shock protein and TPR, dissociate. So they come off of the receptor. The receptor dimerizes, that is, two of them bind together. That they translocate into the cell nucleus, and they bind to specific regulatory site on certain genes within the cell nucleus that are sensitive to glucocorticoids. And by doing so, this hormone receptor interaction with these regulatory sites causes a change in gene expression. So in many cases, the gene is expression is increased. So this is RNA polymerase, RNA polymerase and the accessory proteins involved in transcribing the genetic sequence, the gene sequence to messenger RNA that then codes for the synthesis and expression of specific proteins by that cell. So, so the main mechanism of action of glucocorticoids, cortisol or others, is to affect gene transcription. And what genes are affected and what cells are affected by the hormone depends on a couple of things. First and foremost, does the particular cell express the receptor? So obviously, this is obvious, for a cell to be sensitive or responsive to glucocorticoids, it must have the receptor. And then different cells will have this regulatory sequence, the glucocorticoid, we call it glucocorticoid response element, GRE, will have the sequence on certain genes and not others. So some cells, glucocorticoid will turn on a particular gene. In a different cell, that gene might not be turned on by glucocorticoids if that gene, this regulatory sequence is not active in a different cell. So it's level of the genes and level of expression of the receptor that determine the cell response. So notice that because of this, this takes time, maybe a number of minutes before the messenger RNA levels start to be noticeably or significantly increased, and then still more time before you have a buildup of the levels of the proteins that are being translated here. And so this all takes a certain amount of time, some number of minutes, sometimes hours, depending on, of course, the gene, the protein, et cetera, et cetera. Much more slow kind of response compared to neurotransmitter signaling that occurs on a sometimes sub-second or a millisecond time frame. I should mention before going on that there's also evidence for membrane receptors for glucocorticoids that can mediate fast cellular responses. But the classic responses are occurring here in the cell nucleus. So we saw, if we go back here, we could measure as one biomarker of stress-induced activity of the HPA axis. We could decide to measure secretion of CRH from the PDN. Technically, this is daunting because this is just secreted locally. Very, very little of it reaches the systemic circulation. And so if you really wanted to measure this, you mainly could have to do this in an animal where the animal's anesthetized and you're getting access to the blood vessels right here so that you can look at CRH release. And this has been done, can be done. But for our purposes, since this is almost never done in people, we're going to disregard that. And we're going to start at this level here, looking at ACTH. So ACTH, you're not going to get any significant amounts of ACTH in a saliva sample. So here you pretty much have to be drawing blood. And you can measure ACTH in blood samples, either under baseline or resting conditions, non-stress conditions. But you can also obviously use this as an indicator of the stress response for acute, especially acute stressors. ACTH starts to shoot up fairly quickly. CRH, of course, goes first, ACTH goes second, and ACTH levels in the blood start to increase before you can get measurable increases in cortisol. So if you want to get the earliest responses, again, forgetting about CRF or CRH for the moment, you can go after ACTH. And if the stressor is acute, if it's temporary, then ACTH recovers relatively rapidly as well. So you might imagine that this is kind of like what we were talking about with respect to epinephrine or norepinephrine in the blood. If you want to do this accurately, you might want to have your subjects catheterized in order to get serial blood samples to really be able to follow the ACTH changes in response to stress. Now, I'm going to introduce a term here because we're going to come back to this term later when we talk about cortisol. When you draw a blood sample and you measure something, some biomarker in that blood sample, you're getting a snapshot. This is the level in the blood at that moment in time when you collected the sample. So one term that we use, kind of a colloquial term, is a point sample. It's a point in time. Doesn't mean the moment it was secreted, because something has to be distributed in the, the whole circulatory system. So it's not the moment it was secreted, but it's the moment that it's present at that concentration in that part of the circulatory system, like in your arm, the antecubital vein, where they often, and most typically take a blood sample, we call that the antecubital vein. So that's that point in time. So we're going to come back to that idea later with cortisol as well. There's the kinds of ways that you could measure ACTH are pretty much similar to the kinds of ways that you could measure the catecholamines, the epinephrine and norepinephrine, ELISA, RAA, HPLC, etc. All right, so as I say, sometimes people want to get ACTH. That's fine. Got to get blood samples. Best if they're catheterized. And it's very labile in blood, so you need to make sure it gets chilled very quickly. You may have things in your collection tube to inhibit proteolytic enzymes. ACTH is like a protein. It's a peptide, basically a small protein, can be broken down right in the blood. So there are some tricks if you want to do this accurately, but now we're going to head on to cortisol, which is probably of greater interest to all of you, if you are interested in the HPA axis. So cortisol, for assessing HPA function, obviously baseline, just like ACTH. Diurnal patterns of activity, rhythm. We'll be talking about the rhythm of secretory activity of the HPA axis, and either acute or chronic stress. Here we're focusing on mostly on acute stress, but we'll talk about chronic a little bit here and then big discussion of that later. So obviously we can measure it in blood. Again, point sample. We can measure it in saliva. It's astonishing, before I go on, it's astonishing to me that here at UMass, when I interview undergraduates to possibly do a research assistantship in my lab, and I talk about the HPA axis, and they've had exposure to it either in an neuroendocrinology class or just a behavioral physiology, behavioral neurobiology class or something like that. They've heard of stress, they've heard of cortisol. They somehow had not been told, they didn't know that you could measure this in saliva, despite the fact that salivary, the presence of small amounts of cortisol in saliva was discovered at least 30 years ago and revolutionized cortisol research from that time on, because you don't have to use a needle. You don't have to have people, you don't have to have phlebotomists. You don't have to do it in a doctor's office or a lab or something. You can do it at home. We'll talk about saliva sampling, of course. But the point is, it's really, really important. It was a huge breakthrough in the early 1980s when that was discovered. Urine, cortisol is excreted free in urine or metabolites. Again, 12 or 24-hour excretion. Feces, there's cortisol metabolites that find their way to fecal samples. We hardly do that with people. There's really no particular need to do it with people, but for animal researchers, field researchers, that's highly useful. You're following a troop of chimpanzees or whatever animal it might be, and you are watching them from a distance, and you see an animal poop onto the ground, maybe onto some leaves if they're in the rainforest, and you make a note of that spot, and then later you go and collect that fecal material. And then you bring it back to the lab, you freeze it, bring it back to the lab, and analyze it for glucocorticoid metabolites. So it's very useful, can be very useful in field research. Sweat. Cortisol gets into sweat. It's actually been measured. It's an issue that we'll talk a little bit about when we talk about measuring cortisol in hair and in nails. And various tissues. bodily organs. Of course, we're going to talk about hair and nails and claws. Marine mammal blubber and whale baleen. You may know about whales, certain kinds of whales like right whales, humpback whales that are filter feeders. They swim, they open their giant mouths and take in huge amounts of water as they're swimming. And there's an organ, a tissue called the baleen, which filters out plankton, copepods, things like that. And that's a small fish that serves as their food. And baleen, like hair and nails, has a lot of keratin in it, which gives it its hardness, its stiffness, just like keratin gives nails and hair its brittle quality. And you can measure. Unfortunately, you can't do it non-invasively. So you can only do it if you have a dead whale. But it has been used to assess stress in whales that died, washed up on a beach, etc. And you have various periods of accumulation in these different tissues. So as I said a moment ago, salivary cortisol has largely supplanted plasma measurements in human research. But we'll still bring it up a little bit, because occasionally it's still done. So if you are going to do saliva, which is the same, vast majority of people looking at baseline or acute stress, collect saliva samples. The best way is passive drool. It's what it sounds like. You might have a straw or some way that the person drools, they get saliva out of their mouth, drool it in perhaps into a straw or and then into a collection tube, something like that. That's actually the best way. But you can use oral swabs, cotton type material. One of the first inventors of that was the German company, I think they're German, Sarstedt. That's the Salivet, if you've heard of the Salivet tube. And then Cellometrics came out with their own, I guess that's here, their own oral swabs. And literally a few days ago, I went to the Cellometrics website to see what was the latest that they had to say about that. And they actually presented data showing that using their swab, if you're going to use a swab, was more accurate than Sarstedt, that using the Salivet, you get variable, it actually can affect the assay and you get variable results. So if you're going to do this, I suggest you go to the Cellometrics website, take a look at it. They have a whole section on saliva collection methods. And if you agree with what they show there, then you want to use their oral swabs if you're not going to do passive drool. There are concerns here. You want to make sure that your participants have not eaten or even brushed their teeth shortly before collection. And one of the main arguments against teeth brushing is you want to make sure there's not even very small amounts of blood contamination. The amount of cortisol in saliva, the concentration is much lower than in blood. So small amounts of blood contamination can throw off your value. If you have any worries about that, there are separate tests you can perform on your samples. There's ELISA for ferritin, which is a blood protein. And if that shows up positive, then that's suggestive that you had blood contamination and you might not be able to use the data from that sample. Most people don't do that. They just rely on the fact that their participants are hopefully following instructions and don't have any. Of course, the other thing is if any participant has any dental disease or gum disease and might have some blood due to that, that would obviously be another concern. So things to be aware of. Cortisol in saliva is pretty stable, which is a very, very nice trait. So I know this is a confusing looking graph, but this is the concentration of cortisol in saliva. This is days on this axis. And these are the conditions. So this open, open diamond here is room temperature. So that's shown here. So by, well, this is like 25 days, I guess. This is days here. So, so it's going down here at room temperature. But amazingly, it's still quite a bit of it left even after, you know, many dozens of days. But you don't want to do that, of course. I'm not recommending that. 5 degrees Celsius is here, refrigerated. Minus 20 is here. For some odd reason, this seems to be kind of sloping down a bit. And then minus 80. So these are really, this is within the range of, you know, you see the variability here. So these are really pretty much flat. Cellometrics recommends if you are keeping saliva samples more than, you know, a month or a couple of months, does recommend minus 80 storage. So if you expect to be keeping saliva samples for multiple months, it is recommended, even, you know, despite these data shown here that looks like it should be okay, even at minus 20, they do recommend that. And apparently, if you really, really measure this carefully, there is small degradation occurring over months at minus 20. So again, just to be on the safe side. Oh, the other thing is, very important point, is cortisol survives repeated freeze-thaw cycles. This is a vital piece of information. Think about the idea that you might have some saliva samples that are in your freezer. You take them out to run your Cellometrics assay, ELISA, on the samples, put them back in the freezer, and you run your assay, and oh, some of the data look funky, or for some horrible reason the whole assay went south on you. You have to redo it, or you have to redo a few samples. So now you have to thaw some of those samples again and rerun them. Did you lose cortisol? The answer is pretty much not. Pretty much the cortisol survives freezing and thawing a couple of times. So again, an important thing to know so that you don't have to worry too much about, again, rerunning samples after they've been refrozen. So when cortisol is in your blood, the majority of it is actually bound to plasma proteins. It's not the hormone molecules aren't, most of it is not circulating free. And most of that plasma binding is to a protein that seems to have evolved for that purpose. It's actually called corticosteroid binding globulin, CBG, also sometimes called transcortin. Most of the evidence indicates that only the free, that is the cortisol that is not bound to CBG, is available to get out of the bloodstream into tissues, and therefore has a bioactivity. The binding is not, it's important to realize that this binding is not irreversible. So you can think about the fact that a given molecule of cortisol can bind to a molecule of CBG, and then at some point in time, it unbinds, it dissociates, it comes off. And so you've constantly got a process of binding, dissociation, binding, dissociation. So what we're saying is when you look at a mass of cortisol at a moment in time, where a percentage is bound at that moment in time and another percentage is unbound, the majority is bound. One aspect of this binding is that it's only, we believe, only the unbound cortisol diffuses out of the blood into the saliva. And so that's one of the major factors of why saliva cortisol is much lower than blood. because presumably it's only the free, the unbound cortisol that gets into the salivary cells and gets into the saliva when it's excreted, or secreted, I should say. And lastly, salivary measurement of cortisol would be useless if in fact it didn't track fairly well. So I'm purposely being vague here. The correlation is not perfect by any means, but the correlation is good enough so that we can use salivary as opposed to blood cortisol for things like looking at the circadian rhythm, looking at acute stress responses, changes in baseline cortisol. All those things can reasonably well be done in saliva as a proxy for blood or plasma, even though the correlation isn't perfect. All right, there's even more ways of measuring cortisol than we've talked about so far. So we again have the big three here, ELISA, RAA. RAA is a radioimmunoassay, which has largely been supplanted by ELISA, so that you don't have to use radioactivity. When I was a student and even a postdoc many years ago, we had to measure cortisol by a radioimmunoassay using radioactivity. We don't have to do that anymore because we can use ELISA. But there's also another technique that I just want to make you aware of. It's mostly, currently, it's mostly used for hair cortisol, which is why I bring it up. But of course, it could be done for salivary or blood cortisol as well. So this is liquid chromatography tandem mass spectrometry, or abbreviated LC-MS-MS, which is a very sophisticated analytical technique, requires highly specialized and very expensive equipment. Showing a system here. These guys run for well over $100,000, a typical system new. And like ELISA, you can use it for LC tandem mass spec for measuring, hundreds of different things. Needless to say, though, especially for salivary cortisol, that this would rarely be used, and almost everybody would be using ELISA or occasionally radioimmunoassay. In part because they're commercially available kits. The equipment needed to run these kits is much less expensive and tricky to use compared to that. But another thing that's important to know if you're entering this field or considering to enter the field is that different kits can yield different absolute values. That is, you take a saliva sample and you measure that sample here using kit A, and you measure the same sample in kit B, and the numbers are not the same. So let me give you an example of that. These are data these are not published data. I happen to be fortunate to have had access to these data. So you'll see this afternoon, later this afternoon, when we get into hair and nail cortisol, that my lab for human nail and hair cortisol assays, we use a kit from a company in Ann Arbor, Michigan called Arbor Assays. We use their kit. We used to use Cellometrics, and we still use that kit for animal samples, mainly historical reasons. But a collaborator that I work with at Yale, Monica Ordway, in nursing school at Yale, she collected some saliva samples, and one of the staff people at Cellometrics ran those same set of a large number of samples in both assays. So we had a side-by-side comparison, and this is the result. And here's the actual equation showing relating one to the other. The correlation, as you can see, is very, very tight, but the Cellometrics for just about every sample gave a higher reading than the Arbor assay kit. So how much does that bother us? Well, it's a little bothersome because we can't say for certainty that this, whether it's this value or this value, is the correct, whatever that means, the real value of cortisol. How do we decide, which is which? But the fact that there's such a nice correlation means that if we stay consistent with the kit we're using, and we run a study, and we get certain effects, that relatively speaking, let's say the difference in a simple study, the difference between a stressed group versus a non-stressed group, the difference between the two groups should be about the same, regardless of which kit we used. So that's what we hang our hat on. If we're doing ELISAs or radioimmunoassays, it's not unique to ELISA. This is because the antibody, the assay method uses an antibody, and binding of a substance to an antibody will vary from different antibodies, and an antibody may be reacting to something in your sample besides what you are going after, what you're trying to measure. So there's all kinds of little vagaries there. But be that as it may, and maybe other aspects about how they put the kit together, be that as it may, this is still, it's largely reassuring as long as we understand that the value is going to be kit dependent. It's not kit independent. All right, so one of the things that I mentioned that we can use cortisol, salivary cortisol for is the rhythm. So there's a rhythm, a diurnal means during the day. It's actually a 24-hour rhythm, so it's a circadian circa diem, about a day, about 24 hours. Rhythm of HPA activity. And the nature of that rhythm is that the level of cortisol is high when we wake up. That's here. Then over the next 30 to 45 minutes, for most people on most days, okay, I'm qualifying, but most people, most days, in the next 30 to 45 minutes, it goes up some more. We call that the cortisol awakening response, CAR, cortisol awakening response. That's your response to waking up, getting ready to face the day. Your system is goosing cortisol release a little bit more than it already was at the moment you woke up. Then, so that would typically be the peak for that day. Then it starts to go down fairly steeply in the morning hours, and then more slowly later on. And so it's quite a bit lower at bedtime, and may even get lower as we sleep. And then in the wee hours of the morning, I can't say exactly when, but in the wee hours of the morning, the system starts to activate again. Cortisol levels start to go up from that low level here at night and to reach this point. And it does this every day, day after day after day after day. So if we collect saliva samples at certain key points in the day, usually, not always, but usually people are not collecting saliva in the middle of the night. Usually we're not waking people up in the middle of the night to get a saliva sample. So mostly this is studied as a diurnal or daily rhythm when people are still awake. So if we get, so let's see, if we want to get the CAR, we just need two samples. We get a sample here and we get a sample here. If you really want to look at this in detail, you can get, multiple samples here and see whether the peak's at 30 or 45, whatever. But typically, people get 2 samples here. Then you can collect additional samples during these areas here. You probably want to get one later in the morning, 1 or 2 samples in the afternoon, and then one late evening or bedtime. So, and you can calculate various metrics for the HPA axis, this rhythm, from these various samples. So for the CAR, as I mentioned, it's this. There's a metric that we call the slope. The slope is the difference between wake up over here and bedtime. So if I just want the slope, I collect a saliva sample here and I collect a saliva sample here. Now, the reason I call it the slope, it's kind of weird, but the reason I call it the slope is that you can just draw a line, as they show here, this green line between the two, and look at the slope of that line, the steeper or less steep, more shallow. And you might ask, well, what do you get out of that? Well, we'll talk about that in a minute. You can also get the overall area under the curve. So area under the curve is a good concept to be familiar with, whether it's in endocrinology like here or in pharmacology. If you're doing drug research, for many years I was doing pharmacology of various drugs of abuse. So we might want to inject an animal with a drug and then collect blood samples at various times and look at the rate at which the drug peaked and how it declines. And so literally you're measuring, mathematically, you're measuring the area under the curve of the various time points of your measurements. So here, let's say if you have a saliva sample here, in addition to this one, and this one, it would be defined, the area under the curve would be defined by all of this sort of grayish, yellowish, grayish area here. So we call that AUC, area under the curve. Area under the curve, think about what that gives you. It gives you an indicator, an index of total cortisol release into the blood over that time period, from waking to bedtime. It's not perfect because you haven't measured it literally at every moment in time, but it's a reasonable approximation, a reasonable measurement of how much cortisol was secreted by your adrenal gland that day over that time period, AUC. And you can see here how many saliva samples you need for each of these measures. If CAR only 2, slope only 2, CAR plus slope 3, overall AUC, you need multiple ones in these intermediate time periods, typically 4 or 5. 5 is better, more is better than fewer. Sometimes people even do more than 5. Study we did several years ago to get AUC, we took 5 saliva samples in a group of UMass undergraduates. So let's look at each of these metrics and see why you might be interested in it. So you can actually, I didn't say this, but you can actually measure the CAR not only by the difference between waking to the peak, but also the AUC. So if we go over here, I can measure the area under this whole shaded area here, where it says CAR AUC. So that would be another way as opposed to measuring, looking at the peak. CAR measurements, has been shown empirically, CAR measurement to be accurate. Your participants have got to follow the directions, which means you don't take your first saliva sample 5 minutes after you woke up. You take it like you have your collection materials next to your bed, ideally. And as soon as you wake up and clear your head of the cobwebs of sleep, you get that first saliva sample. And then you meticulously time out 30 or 45 minutes, whatever you've been asked to do for your next saliva sample. If you don't do that, it's a very noisy and inaccurate measurement. There are ways of doing that to try to ensure participant compliance, which we don't have to get into right now, but otherwise you're just taking their word for it and may not be good. Importantly, the CAR is affected by various psychosocial and other factors. So chronic stress can affect the CAR. Psychosocial variables can affect it. I'm going to show you a study in a moment, in the next slide. It's also been related to certain psychiatric disorders such as depression. So here's a study. I kind of like this study. The head of the lab is, what's his name? Because I'm blocking on his name at this moment. He's down in Australia. Sarnier is his last name, Zoltan Sarnier. Yeah, so his group compared, this is a small n. Most of the time you want to have bigger sample sizes than this, but he still got significant effects. He had 11 indigenous students in Australia, at an Australian university, his university, James Cook, as well as 11 non-indigenous. These were adult education students, so they weren't a typical college age. And they measured cortisol at 3 time points, wake up, 30 minutes for the CAR, and then 12 hours later to get the slope. And you see several things right off the bat just by eyeballing the data. You see that the indigenous group had a significantly lower baseline cortisol at wake up. So their wake up level was blunted. Second of all, hardly any CAR, hardly any difference between the cortisol value here and the cortisol value here compared to the non-indigenous. And then the slope, if you mentally draw the line between here and here and here and here, you can easily see there's a big difference there. So the way I think about it, rather, we call it the slope, but it's really the amplitude. It's the amplitude of the rhythm from waking to late day. So these are not good things. Having low cortisol, just like having excessively high cortisol, having low cortisol, blunted stress responses, blunted CAR, is indicative of dysregulation of the system. So this is not a good thing. So of course, indigenous peoples in Australia are subject to a lot of discrimination and a lot of differences in average socioeconomic status and so forth in those populations, even now, even after many years of trying to deal with that. So here's, I like this study, relatively recent one. There are 2 studies here. The data is actually here, and then a subsequent study I mentioned here. So this was a meta-analysis looking at a bunch of different illnesses and disorders listed here. Here's the lower limit. This is the correlation between the waking value and the later value as a way of measuring the slope, and then calculating z-values. So here comparing people with the disorder against matched controls without the disorder among a number of studies so that you have a meta-analysis. And so here's each disorder. You can see all of them except for anxiety is in the direction of a flatter slope in people with the disorder compared to healthy controls. Anxiety is the only, disorders are only the one in the opposite direction. So in general, having a flatter cortisol slope is associated with a tendency towards illness. And second study, more complicated, so I'm not showing the data here, are that the flattened slope is especially likely to occur in people who have a combination of both early life stress, meaning childhood stress, or current life stress. Okay, so so we've looked at CAR, we've looked at slope. What about stress responses? The obvious another way of using salivary cortisol. So we're coming back to our Trier social stress test. And there's a version of the test that was designed for groups. So you can test more than one person at a time. So this is the TSST-G. And this is a good thing to know. So many participants, as you would expect, many participants do respond with a good cortisol response to the Trier, but some don't. So here we see, this is salivary cortisol here. This is minutes in relation to the test. Here's the test period here. -1 minute before the test starts, 1 minute after the test ended, and then recovery. So here's controls, no stress. That's these guys here. Responders are over here, nice, robust response, and then non-responders. Now notice, how do you get something like this? How do you figure out that somebody is a responder or not? This is post-hoc. This is a post-hoc separation of the groups. So in other words, you get your group data, and then you look at your group data, and you say, wait a second, they're not all showing the same pattern. Some people are showing a response, other people are showing not a response. You set up some criterion for determining what's a response. You have to do that, of course. And then you come up with this. Notice that heart rate, here's heart rate, is the non-responders here don't respond as vigorously in terms of their heart rate, but they do show a response more than the controls. So even the controls show a little elevation in heart rate just by being in the lab, coming in and doing whatever they had them do. But so these two don't completely go together. Cardiovascular response to stress and the cortisol. Time of day. If you have a choice about when to administer a stressor, whether it's a Trier or any other, your best bet, if you want to get a robust response, is to do it in the morning. And this surprised me actually, because I would have thought, if I didn't know these data, I would have thought that maybe when the cortisol levels were lower in the afternoon, according to the rhythm, that maybe you'd be more stress sensitive when the baseline, your starting point, was lower, but it doesn't turn out that way. So here's the morning, here's the afternoon, so here's where you are in the rhythm. Morning test, afternoon test, and it looks like the magnitude of the rise is not much different, but the AUC is significantly different. So suggestive that, if possible, you want to do your testing in the morning. One of the problems with any laboratory stress test, whether it's Trier or any other, is what we would call anticipatory stress responses. Think about the fact that you're disrupting the person's normal routine. They have to come into your lab. They know you're going to do something to them. You may not have disclosed exactly what it's going to be. You probably haven't, in fact, ahead of time. You debrief them afterwards, obviously. But so many people will respond to the any worry, anxiety, anticipation of whatever is going to be done to them with at least a modest stress response. So this was shown empirically in the study by Engert et al. So here are the groups. Some participants were told at baseline, that's here, that they be given the stress test. So in this case, they were told about it, but the test was omitted. So that's these guys here. So see, they're told what's going to happen, and you can see that so this is about 10 minutes before this, by about 18 minutes after the disclosure that they're going to be tested in a stress test, they show a modest but measurable cortisol rise in their saliva. Others were informed about the stress test just 1 minute before T0 instead of the usual 10 minutes. That's the stress only. And you can see the same rise here, but this is the pure stress rise and not the anticipation. We've gotten rid of the anticipation over here. So if you look at this versus the peak here, you see that if people were, well, they'd be stressed over here, I guess. They'd be stressed over here. But you can see that this anticipatory stress response is going to interfere with your measure of the response to the stressor itself. Okay, so this one I'm just going to skip over. It just shows that there are different, what they call reactive responders as opposed to anticipatory responders and non-responders. I'm going to skip over this one so we can keep things moving along here. All right, this is important. I mentioned that you can get both cortisol and alpha amylase in the same saliva samples. The problem with doing that, though, is that the timing of the time course of the response is a little different between the two. And so you need to think about your times of saliva collection to give you the best possible ability to look at both. So here we see where lots of saliva samples were collected at all these different time points. And amylase, we see responds more quickly. This is the 0 time point here, onset of the stress. Amylase is responding more quickly. Cortisol is more delayed. And so amylase is also recovering before cortisol is recovering. So you would think about, and you'll see in the literature, I mean, you don't have to look at this graph and figure it out on your own. If you look in the literature, you'll see what various researchers have done in terms of their timing of saliva collection to try to optimize as best they can the measurement of both biomarkers. Some of you may have heard of this test if you are familiar with various tests of the HPA axis. This is called the dexamethasone suppression test, and it's specifically a test of the negative feedback properties of the individual's system. So dexamethasone is a synthetic glucocorticoid. It does not get very effectively into the brain, so it's mostly working at the glucocorticoid receptors in the pituitary gland. Pituitary gland is not protected by the blood-brain barrier, so the pituitary gland is subjected to circulating substances, drugs, hormones, etc., that may be at least partially restricted or reduced from entry into the brain, which includes dexamethasone. Anyway, it's used medically. It's a very powerful anti-inflammatory drug, so it is used medically for that purpose. The important thing here is that it activates the negative feedback loop. again at the level of the pituitary gland. So when you're performing this test, there are two different ways you can measure the response. Again, because it's shutting off the negative feedback loop, it's causing cortisol levels to decline. So it's binding to the receptors in the pituitary gland. Pituitary gland shuts off ACTH. Adrenal gland shuts off cortisol. And so the levels start getting lower and lower over time until the effect wears off. So you can either look at the degree of suppression, how low does it go? Or in many cases, researchers look at, or doctors, sometimes it's used diagnostically, the time needed for resumption of cortisol release. One of the ways that it's been used in psychiatry is to assess negative feedback dysregulation in patients with PTSD. So here's an example of that. So here's a healthy control group, and here's a group of individuals diagnosed with PTSD. They're using plasma cortisol in this case. And here's the baseline. Both groups have an average baseline level that's about the same. And then at some time point, I don't know the details of this, at some time point, they looked at the plasma cortisol levels in the two groups, and you can see that the individuals with PTSD actually had greater suppression. If you think about what this means, it means that their negative feedback system is actually hypersensitive. It's reacting more to the dexamethasone by suppressing ACTH and thence suppressing cortisol even more than in a control group. So negative feedback dysregulation is interpreted to be an indicator of either altered expression of the receptor. It's possible that the pituitary gland is expressing more receptors, or signaling how the receptor is affecting downstream cellular mechanisms that work to suppress ACTH release. Either one could be happening. Okay, we're almost ready for our break. So an interim summary. We've been focusing mostly on saliva, salivary cortisol, the various uses of that. As we talked about, it's mainly used now as an alternative to blood for point sampling. A lot of different things you can use it for, so it's very important if you want to study this system to be familiar with measuring cortisol in saliva and the best ways to do that. And we also saw that when you're using antibody-based methods like ELISA or RAA, radioimmunoassay, if you're doing that, that the absolute values can vary depending on your kit or other aspects of your method. So let's get some questions and then we'll have a break. Questions?
I have a question. What is the evolutionary purpose of the CAR? I mean, if
yeah, well, if it exists, yeah, presumably it arose during evolution. This we can only speculate about that. I think, and there are different speculations. I think one of the main ones is that by elevating cortisol even more, that it's preparing your physiological systems for dealing with the events of the day. Why it goes up after you're awake and not already at a peak before you wake up, I'm not sure, except that it probably allows, by having it I'm speculating, by having it occur, that is this additional rise after you wake up, allows for some regulation that may occur during sleep. Or, well, yeah, I guess that's probably the best thing to say is what may be going on during your sleep. Keep in mind that, you know, we now know from a lot of research, including some even going on at UMass in my department, that sleep plays a very important role in consolidation of learning that you've had during the preceding day. So one could imagine, one could speculate that the effects of the previous day's activity on your sleep and what's happening during your sleep could have some impact on the CAR in the following day that perhaps has some as yet unknown adaptive value. That's the best I can come up with, because the answer is we really don't know. So we just have speculation about possibilities.
Thank you.
Sure.
I also have a question on cortisol. I know, like, when we started in this slide, you said about you can measure chronic stress or acute stress using cortisol. So, like, I'm just like wondering how specifically I will know that this was detecting a chronic stress rather than the acute stress that was available. Like, is there a way of like kind of like just very specifically like capturing one of the other?
Well, first of all, I think it's really important to realize that cortisol, or even if we say elevated cortisol, is not equal to stress. It's not stress equals cortisol. Cortisol is a hormone secreted by the adrenal glands that is stress sensitive. And in properly controlled experiments, changes in cortisol or differences in cortisol between groups, or changes if you're doing longitudinal studies, can reflect the influence of stress or stressors on that system. But it's not so simple as, you know, when you read in the newspaper or magazine article about somebody, you know, measuring cortisol, and the science writer or the reporter more or less assumes that cortisol equals stress. Well, it's not. That's way oversimplified. As far as acute versus chronic stress, the salivary cortisol, because of the fact that it is subject to a rhythm that alters the level of cortisol over the course of the day a lot, plus the fact that it's sensitive to any minor stress that you might have had before you went to the experiment, an argument or a bad night's sleep the night before, whatever. Salivary cortisol is not best suited. It's not so easily suited for looking at the effects of chronic stress. And that's why the remainder of the day, we're going to be talking about cortisol in hair and in nails and things like that as possible markers of the effects of chronic stress on the HPA axis. So, but I don't want to say more about it right now because I'm going to be repeating myself. I'm going to talk about those issues after our break.
Okay, thank you so much.
Yeah. Mm-hmm.
I had a question about the stability of the salivary cortisol rhythm, across the life course. So if there was a child with who experienced a lot of adversity and had more of a flatter slope, does that tend to be pretty stable into adulthood, or do we see that it changes just based on experience?
Yeah, I'm not familiar with. I don't know I don't know if there have been specific experiments to try to test that. Think about the complexity of doing a study like that, where you're doing that long a time span and getting that funded by the NIH. No, I mean, even regardless of the funding issue, which we all know is complicated by itself. But yeah, it's trying to follow kids, individuals from childhood all the way into adulthood to see if those effects are still present. There are some studies doing that sort of thing. I don't know if any of those studies have specifically looked at whether effects of things like childhood adversity on slope, if they're manifested in a particular cohort of kids, whether that persists into adulthood. I can't say, I don't know.
Okay, thank you.
Interesting question. Other questions? Okay, so it's 2:15. So we're going to take a 15-minute break and start again at 2:30. All right, so now we're going to segue, and this is really getting into now my lab's specialty. So I hope any you guys have any tricky questions. Hopefully I'll be in a good position to answer them. So let me start sharing my screen again. All right. So there have already been several questions from you guys about, what if you want to study some kind of a chronic stress situation, and do you do that the same way that you study acute stress? How do you separate if somebody's under chronic stress and you're looking, for example, at their salivary cortisol? How well do you know whether that's from their chronic stress or some acute stress? Sorry, it's there's a lot of talking from me today, so I've got some water here. I'm gonna try to keep my voice going and not lose it. So the reasons, the first slide lays out the obvious reasons why we're interested in chronic stress. And even though this workshop is treating cortisol and these other measures as biomarkers, cortisol is really important because it not only is a marker for chronic stress, it's a mediator. It actually is involved in the pathway or pathways between stress and ill health. So that makes it especially important. So in chronic stress, up until the mid-2000s, most researchers who wanted to use HPA axis, use cortisol to study situations of chronic stress, still pretty much had to rely on salivary cortisol. And many studies are out there, and people do continue. I'm not trying to totally diss salivary cortisol, even for chronic stress. Many researchers continue to use salivary cortisol, even for chronic stress studies. But it's not ideal for a number of reasons, some of which we've already discussed. Sensitivity of salivary cortisol to rhythm, to a stressor of that occurring that day, and so on and so forth. So in this literature, in this early literature on chronic stress and salivary cortisol measurements, the results have been highly inconsistent. And I don't want to imply, before I go on, I don't want to imply that it means that now that we're using hair cortisol, that everything is all consistent now, because it's not. So I'm not trying to say that we've solved that problem. But nevertheless, talking about using salivary cortisol, some researchers found that groups, again, cross-sectional studies, groups that were deemed to be under chronic stress compared to a control group, the stress group showed increased HP activity. Some studies showed decreased HP activity, and inevitably, some studies found no difference between the two groups. So, you know, it's like so many areas of science, you get these discrepancies. And so, you know, what the heck is going on? Can we make any sense of it? Well, we haven't totally solved the problem, but this is a very instructive paper. If you aren't aware of this paper, I strongly recommend that you read it. It is in my source files by Miller and other authors. And as you can see, this was a meta-analysis of 26 studies that used various measures of daily output. So ideally, it would be a detailed AUC, like we discussed, but many studies did not use that. You might have used urinary, might have used slope. So one of the problems of this analysis is that there were different ways in different studies of measuring daily output. So the authors for the meta-analysis tried to standardize this so you could compare one study against another. And each circle represents one study in the meta-analysis, and the size of the circle corresponds to the weight of that study in the analysis, which is more or less the number of subjects in that study whose data contributed to the meta-analysis. And the key variable here in this graph, there's much more to the paper than just this, but this is what I want to focus on. You can see that this is a log scale. The key thing is that when they looked at the relationship between an estimate of daily cortisol output with time since the onset of the stressor, there was a negative relationship, an inverse relationship. So the more time since, when you look at all the data overall, the more time since the onset of the stressor, the lower the cortisol output. And here, the zero value here would mean no difference between the, no change in cortisol output. So there's a zero here, which corresponds to roughly here on the line. So these are studies with positive effect, more cortisol output, and after the zero, down here are the studies with a negative. And this is the best fit line. So what the authors argue, and this has been accepted at least as a hypothesis in the field, not proven, but hypothesis, because these are, after all, just correlative data, and you can see they're quite noisy. Many other factors go into these results. But the authors hypothesized that when you have a major ongoing stressor, initially and for some period of time, the HPA axis is overactive. So you're putting out more cortisol. You have a hyperactivity of the system. Over time, as a counter-regulatory response, which would fit with, by the way, allostatic theory, allostasis. Over time, the system starts to adapt to this constant overactivity, and it starts coming down. Maybe for some people, in some situations, it reaches sort of a null point here, where as a group, there's no difference. Counts for the studies of no difference, at least some of them maybe. And then if it keeps going, you get a blunting of HPA activity. Okay, everybody with me on that? All right. So here's a study a recent study that sort of tapped into this a little bit. So the previous graph was from a meta-analysis of many studies. Again, you could see a lot of differences in methodology between studies, a lot of noise. This is a single study in which the researchers assessed both cumulative life stress using strain. Remember strain, that was one of the measures we talked about this morning. So it's a lifetime subjective measure of lifetime stress, or I shouldn't say subjective, self-reported lifetime stress. Here's the age range. So these are young to late middle-aged adults. And they also looked at the acute stress response using the Trier for groups. They collected saliva at baseline and 10 minutes post-stress for cortisol and DHEA. I mentioned earlier, there was a question about DHEA. And I'm only going to present the cortisol data, but I'll mention what happened with DHEA as well. And to look at the relationship between lifetime stress and acute stress response, they, first of all, they log transformed the hormone concentrations. It's not unusual. This is true not only for saliva, but for hair cortisol. The distribution of the data is often right skewed. And so you need to do some kind of transformation to normalize the distribution. This is one of the common ones. And they looked at the change in hormone levels from baseline to 10 minutes post-stress. They didn't have detailed measures here in the Trier, so that's a limitation. And then they correlated the hormone responses with the strain scores. So each one of these is an individual subject. Here's the best fit line, confidence interval. This is the cortisol data. Log transformed. Life stressors, this is standardized. So it's not literally the score. And what we see is, and it looks something like that previous one, even though the other was different. It wasn't stress, it was daily cortisol output, but it's a similar idea. So more lifetime stress caused, or is related to, maybe caused, possibly caused, a reduction overall, on average, a reduced cortisol response to the acute stress. Interestingly, DHEA went the opposite direction. Again, if you look up the paper, you'll see the graph for DHEA, it's the opposite direction. And so there's a similarity. I was struck by this because there's a similarity with the phenomenon of, but this time it actually looked at stress over a whole lifetime up to the time of the study. And so maybe they're telling us two different sides of the same coin of how cumulative stress in some way, in some period of time, can lead to the system becoming blunted, measured either as total cortisol output over a day or days, or a blunted stress response in this case. So going back to the idea of, the old way, the traditional way before hair came into vogue, if you will, of trying to measure chronic stress, HPA axis responses to chronic stress using saliva. Your biggest problem is that salivary cortisol has only a modest trait-like quality. So, a lot of you are psychologists or in other aspects of a science that has a social aspect to it, social science. I like to consider psychology properly done as a biological science, but traditionally it's a social science. Anyway, so we know that traits are sort of enduring qualities of an individual. We talk about a psychological trait. something that is relatively consistently manifested in different contexts or different situations. So one person might be relatively outgoing, so they're outgoing in many situations. Another person is shy, so they tend to be shy and withdrawn in many situations. So those are traits. So again, relatively consistent, relatively enduring. So we can look at biological markers in the same way. To what extent is a biomarker have a trait-like quality? And this was actually a number of years ago, again, Kirschbaum's group, they've done so much wonderful work looking at, assessing mathematically the trait-like quality, what they call latent trait of salivary cortisol. So they did this by taking 2 measurements 6 weeks apart. And 75%, well, 21% of the variance was due to the latent trait. So only 1/5 of the variability in salivary cortisol levels can be explained by a consistency of that variable. 75% of variance from situational factors, something that happened on that day or recently, and then 4% they estimated from measurement error to make 100%. So that's relatively modest, which is another way of saying it's noisy. It's not terribly stable. So the way we think about this is that if you want to measure like that AUC or slope or CAR, any of those things, you need to get multiple days of measurements for each subject. If you do it on one day, it's pretty crappy. Pardon my French, but you're not going to get good data if you measure any of those things from your participants on one day, unless you have a huge pool of subjects. So you need to do it over multiple days. And if you want to get cortisol output, which means AUC, and you're doing like 5 measurements like we did for our study of nursing students, we did it on 3 separate days. Some people, so that's 15 samples for each person to get an average AUC or representative AUC from that person. Some people do 4 samples. Oh, I'm sorry. Well, some people do 2 days. They argue, well, 2 days is better than 1. Yeah, but 3 is better. Some people argue you even need to do more than 3. We chose 3. It just gets to the point where how much time and effort are you going to put into it, right? So, so if that's what you have to do in a chronic stress study to find out whether your stressed, presumably stressed subjects are putting out more or less cortisol than your control group, look at how many samples you have to analyze. That's what I want to leave you with as one of the takeaways here. So is there a more efficient method? And of course, the answer is, since I'm giving this presentation, and one of the developers of this method is yes. And that is measuring cortisol that accumulates stably in a tissue that's readily accessible and that the cortisol can be extracted and measured. And most of the research up to now, most of the data that we have up to now is using hair, which is where this technique was pioneered. We were not the first people to measure cortisol in hair. As I'll tell you momentarily, in a few minutes, we were the first to show that it's sensitive to stress. And that was one of the big contributions of our lab. But nails, we'll talk later a little bit about nails. It's more of an emerging field because there's a lot less known about it than hair. Anyway, so the idea is that hair grows, nails grow over periods of weeks to months, and cortisol accumulation in those kinds of samples, in those tissues or materials, whatever you want to call them, represents, it's like the AUC every day. It's like every day's AUC, and then collect it over many, many days, over weeks, even months. That's how we think about this measure. So you can see the potentially, if this is right, if it's doing what we think it's doing, you can see the power of this approach. So the idea of measuring hair, a cortisol in hair is really not terribly dissimilar from hair drug testing. You might not know, we discovered this when we started doing this work a number of years ago. We discovered that there's an organization, an international organization called the Society for Hair Testing. It had been in existence for a number of years. When we found out about it, it still exists. And it mostly involved with forensic analysis in hair samples, although it's also applicable to hormones, as in our case, also environmental pollutants. So heavy metals can get into your hair, lipid-soluble pesticides that are in the environment can get into your hair. Anything that can get into your bloodstream, whether it's a drug or, like I say, a pollutant or a hormone that your body is making, can get into your hair and potentially be measured. So this is just, I just downloaded this from a website from a company that does hair drug testing. So if you're an employer, you might want to, employ that company to, if you want to test job applicants or your employees for drug use. So our first study, we're actually doing research with my collaborator, Melinda Novak, collaborator and wife, who had been doing research for many years at the New England Primate Research Center, which was outside of Boston. Actually, it's closed now, but it was being administered at the time by Harvard Medical School. And I'm not going to get into the whole rationale of what we were studying, but we were looking at captive adult male rhesus monkeys. This, of course, is a monkey in the wild, not at the New England Research Center, but it shows what an adult rhesus monkey looks like. And we had reason to wonder whether the monkeys that we were studying compared to controls that we also were looking at might be under differential stress, specifically chronic stress. And we had the idea, I'm not going to get into, we don't have time to get into where we got this idea, but we did get the idea of seeing whether we could measure cortisol in their hair, body hair. So we developed, we tested a number of different methods for preparing the samples. We worked out a method for extracting the cortisol from the hair. We measure the cortisol by ELISA. We did what we call in immune tests, something called a parallelism test, which is a test for validating that the antibody that you're using is reacting to the analyte, what you're trying to measure, and not possibly something else. And then the big thing was whether the hair cortisol levels were sensitive to a stressor. So as you can see, the paper was published in 2006 in General Comparative Endocrinology. It's in the, of course, folder of papers. So these monkeys had been living in, were living in stable housing conditions for a long time. The Primate Center had funds from the government, from the NIH, to build a new building. And our monkeys had been in an old building, and they were going to be relocated to new rooms. Now, these animals were all in individual pens. They were in large floor-to-ceiling pens. Each animal was in its own pen, a lot of room to move around. Individually housed, they're adult males, so they can be dangerous. They can fight with each other, so in a way it was good that they were each by itself. But they can also communicate with each other. It's mesh cages. They vocalize, they threaten, they can bark, show their big teeth, and so forth. So there's a lot of interaction, behavioral interaction going on between animals in the same room. So the new building was going to have not only a different room, of course, but smaller cages, still permissible size. There are regulations on this, but also a lot of new roommates. A lot of animals that these animals were not familiar with, not part of the group that we were studying. And we thought that this move, this abrupt change, might be very stressful. and that the stress might continue for a substantial period of time. So we obtained saliva, plasma, and hair samples before and after the move. Of course, I'm not showing you all the data, they're in the paper, but plasma sample, cortisol went up, saliva cortisol went up, and the important thing is this graph here. So this post-move was 2 months after, this is the baseline sample just before they were moved. They were anesthetized. We shaved some hair from the back of their neck. And then 2 months later, they were anesthetized again. And we shaved hair from the exact same place. And this was really important because it meant that the hair that we measured cortisol in here had to have grown during that 2 months since they had moved. And then we shaved the same spot again a year after the baseline. And you can see what happened. There's a beautiful study. Virtually every monkey, every single monkey went up from here to here. You didn't even have to run any stats on it. You could have just, I don't have the individual data plotted here, but it was just about a uniform response. So this was the first, as I said, it wasn't the first demonstration, even in humans. A few years earlier, a group had published measuring cortisol and cortisone, metabolite of cortisol, in human hair samples. But it really hadn't made much impact in the literature because what did it mean? What could you use it for? What this showed was, look at that, an ongoing stressor. And by the way, the animals were behaviorally affected for a long period of time. Their sleep was disrupted. So we had many, many measures showing that these animals were persistently stressed, even out beyond this 2-month time point. So that was besides the methods development that we had done, this was the big thing. And it opened up the field of using cortisol deposition or accumulation in hair as potential biomarker of chronic stress. So sampling of hair in animal studies, and our lab up to now has looked at many different species. We've measured cortisol obviously we're doing a lot of human work, many different species of non-human primates, pigs, goats, cats, dogs, polar bears, interesting story about that. And we may be doing cattle soon. We were contacted by a researcher at Texas Tech who's running a study or wants to run a study with cattle and calves and maybe sending us hair samples from that. So there you decide where you want to collect the hair. For the monkeys, it was the back of the neck. For human studies, the best place is what's called the posterior vertex, which pretty much shown here. This is a little bit lower than what I would call the posterior vertex. I would say that's really up a little bit higher. It's the occipital region of the skull. And what you want is to cut the hair closest to the scalp. And you want to make sure that you know when you cut this piece of hair and you have these long strands of hair, make sure you want to know which end is that. Because, and I'll explain why in a minute, but that's vitally important. So you're looking, remember the hair is growing. We believe that cortisol is only deposited into the hair as long as the hair is growing. Hair goes through stages. The follicle cells that make hair go through a life cycle. And so the hair grows, and at some point the growth stops, and then the follicle discards the hair. It's called the telogen phase. And so that's when, like if you're brushing your hair or you're in the shower or something and you see some small amounts of hair, that's probably just telogen hair that just was discarded from some follicles on your head. It doesn't mean there's anything wrong unless you're losing a lot of hair, that's different. But anyway, so what we want to know is what are the features of hair growth in people. So the hair growth rate is averages, and this has been shown empirically, averages about 1 centimeter a month. Now, it's really important to say average because it can vary in the same person. It can vary over time. It can change with nutritional status. It changes with pregnancy, changes with age, especially children. There's differences in ethnic groups, especially African Americans versus Caucasians. There's differences in growth rate. So this 1 centimeter a month is for, the average is for a white European origin, American or white European. But even then, even despite individual differences and ethnic differences, there's a lot of differences just from person to person within a group. So this adds noise to your data because we almost never know an individual subject's growth rate. So we just have to use the average value. So this means that if we go back to the previous slide and starting here, close to the scalp, if we take a ruler and we measure out 3 centimeters and then make a second cut over here, and we use that piece of hair, that the cortisol that we extracted from that piece of hair represents cortisol output roughly during the previous 3 months. If it was 2 centimeters, if we cut it over here, then it's roughly the previous 2 months, and so on and so forth. So we think that the cortisol is relatively stable, doesn't move too much. There has been some data that might challenge that, but for the most part, we believe that the cortisol is fairly stable in the hair, doesn't move around too much. Hair has a very low water content and low lipid content, which would tend to reduce the rate of movement. But nevertheless, there could be some, but we're going on the assumption that it doesn't move too much. But this allows segmental analysis. So you could cut a hair into segments, maybe 2 centimeters and then the next 2 centimeters, or even 1 centimeter, the next centimeter, and get a calendar of cortisol output by month. It's a little dicey doing that for a couple reasons, but it has been done. It is done sometimes. If you really get into the literature, you'll see studies doing that. So it is possible. I mentioned, I think, that cortisol, we think it only accumulates in hair during the growth phase. And like salivary cortisol, we think it reflects the free fraction. So just like cortisol has to diffuse into the salivary cells, those acinar cells to get into saliva, it has to diffuse into the hair follicles, the cells in the scalp and the skin that make the hair shaft. And the hair shaft is extruded from the follicular cells. So the cortisol diffuses, the free cortisol diffuses into those cells. One of the beauties, unlike salivary cortisol, is that hair cortisol doesn't appear to be affected by an acute stressful event that occurred before sample collection. Let me give you empirical evidence for that. This was a study published in 2015. They're actually, among other things, they were looking at cortisol and sweat. But what I want to point out here, I'm not even giving you the sweat data. What I'm focusing on is salivary cortisol here and hair cortisol here. So because they wanted the subjects to sweat a lot, they put them on a treadmill for 30 minutes to induce sweating. So here is, they took saliva samples at these various time points. So here's baseline before. For some reason, the treadmill group, this is the intervention, is the quote treadmill group, had started out with a higher baseline than the non-treadmill group. Don't know why, just random variation. But you can see a nice salivary cortisol response. Physical exercise acts as a kind of a stressor. So you do get a cortisol response to physical exercise. No change here. But now look at the hair cortisol in the two groups. So here's the again, there was a difference in baseline for some reason, but the important thing is that the intervention group that showed this nice cortisol response, the hair was measured. Oh, I'm sorry. This was 5 minutes before the treadmill, and this was 5 minutes after the treadmill, and then this was 24 hours after the treadmill. So point is that the treadmill produced no change in hair cortisol despite the increase in salivary. So it means that you can more or less not worry about whether your subjects might be stressed to some extent when the hair collection is going on. Based on these and other data, we don't think that it's a significant problem. The other thing is that hair cortisol has better trait-like features than salivary cortisol. I showed you the latent trait was estimated at about 21%. So here's some data. Hair cortisol, and these participants were sampled either twice at a 1-year interval. That's study 1 here. Let's look at that one first. So this is 1 year apart, same participants. Each dot, of course, represents the hair cortisol at, this is time 1 here, 2009. Time 2 is here, 2010. And you can see that there's a very strong correlation, 0.73, between the hair cortisol values at the 2 different time points. So interestingly, it doesn't go through 0. So something overall was changing from one time point to the other, but to whatever extent there might have been a change, it occurred to the same extent so that the 2 were well correlated. And then study 2 is just a little different kind of experimental approach, but it showed the same thing. So again, you can see correlations of over 0.7 or here nearly 0.7. So, so this tells us that we have data of our own also indicating a pretty good stability, assuming that people are living under stable conditions, obviously that there's no big change in their stress. Now, I mentioned that you might want to collect, like 3 centimeters of hair for 3 months. What if you want to go back 6 months, 9 months, a year? If you have people with really long hair, typically women, not always, but if somebody has really long hair, maybe you could go back, as far as their hair will let you go. Can you do that? And the answer is no. There is a limitation. And different names have been put on this. We use the term washout effect because we think this is mostly from exposure of cortisol in the hair to repeated washing and shampooing. But there is also an effect of exposure to UV radiation. This has been shown empirically. Washing and shampooing, we believe, leaches. It's pulling some of the cortisol out of the hair. Hair swells in water, and the cortisol may be more likely to diffuse out. UV radiation actually breaks down cortisol molecules energetically. So if you look at the first demonstration of this, this was the first demonstration, Kirschbaum's group again. So they had women who had quite long hair, N of 9, and they took 3 centimeter segments. So this is the 3 centimeters closest to the scalp, next 3 centimeters further out, and so on. And it seems to asymptote out here, way out. But because of this, we would believe that it's not smart to go much beyond this 3 months here, the 3 centimeter segment. So for most of the studies that we're involved with, it's either 3, maybe pushing it to 4 if there's really good reason to do so, or less. Now notice, not all studies have observed this washout effect. So, but I think it's consistent enough that to take it into account. Whoops, sorry, I didn't mean to do that. All right. Oops. Okay. So there's another limitation. I'm presenting this because I really want to make sure you guys know about this. This is something that I have to say is rarely discussed in the hair cortisol literature. It's rarely talked about. People, experts in the field know about this, and we know we have to keep it in mind. And so now you guys are going to know about it. And I'm going to address the question of how much is this a problem or not. The issue is that there's quite decent evidence that within the hair follicle, again, this is the organ that's making the hair shaft that you're measuring cortisol in, there's local cortisol synthesis, meaning that this can be a source of cortisol that's in that hair that you're measuring. Now keep in mind that the whole premise of this biomarker is that you're measuring cortisol that was in the blood, that came from the adrenal glands, and that is sensitive to stress, especially chronic stress. So what's this all about? And how do we need to think about it? So here's the first, to my knowledge, the first evidence of this. And so this is, so there's more research beyond this paper, quite a bit more, indicating that the skin is in fact an endocrine organ, not just your standard glands. But here we're focusing on the hair follicle, that part of the skin, for those cells in the skin. And there's evidence, so here we see this is immunofluorescence, immunofluorescence staining for ACTH. So each of these is a explant. So this is taken from human skin and cultured. And so this was treated with CRH, and you see ACTH staining goes way up. So ACTH in these cells is sensitive to CRH. We'll forget about that one, we don't have to worry about that one. This is the receptor for CRH. And we see that the receptor is localized just to this band here, but there is evidence for the receptor. Here's cortisol, sensitive to ACTH. Here's cortisol staining. Here's the glucocorticoid receptor. So the whole darn axis, the whole HPA axis is like present in your hair follicle. It's crazy. And it shows the expected responses to stimulation. I also might add, I'm not showing the data here, but they also showed that cortisol was released into the Petri dish or whatever these explants were cultured in. So they actually released cortisol. And there's other studies as well. I just wanted to give this one. This was the first, and it shows all these different parts of the system. Now, bottom line is we have not found any way of completely figuring out the contribution of local synthesis to hair cortisol for any sample. For any sample that you might get in your lab or send off to a collaborator, you don't know how much of that came from the blood, the cortisol that they're going to measure, how much came from hair follicles. So that sounds like, if we just left it at that, that sounds like, well, we're really in a muddle But in point of fact, maybe not so bad as it sounds. And this is why. Oh, well, sorry, I got a little ahead of myself. This is just showing the potential mechanisms of cortisol incorporation, focusing on bloodstream. There could also be diffusion from surrounding tissues of the skin. The hair shaft, there's cortisol not only in sweat, I mentioned that, but in sebum, the secretion of sebaceous glands near sebum. And then I mentioned local synthesis. And then the mechanisms of cortisol elimination. Okay, so that's from a review paper that we published a little while back, almost 10 years ago. All right, so let's get back to this issue of local synthesis. So, question. Does local cortisol synthesis invalidate use of hair cortisol levels? And this is so I raised the question. So I'm playing devil's advocate to myself, raising the question, does it invalidate? How do we respond to that? So here's the data that I think are relevant. First of all, hair cortisol levels are highly correlated with average cortisol output measuring salivary AUC. A couple papers, we've done this ourselves, although we haven't published it. The paper from Short, I think I have it here. Yes. So this is a correlation of 0.6, not bad. This is 1 month. So they measured it over a number of weeks, over a month. The integrated salivary concentrations, AUC, against hair cortisol in the same subjects. So pretty decent, pretty decent. So again, the saliva has to be coming from the blood, right? So it's saying that there's a pretty good tracking of the blood cortisol over time with what we see in hair. That's point number one. Point number two, a very nice meta-analysis. It's in your readings. I highly recommend it by Stalder and co-authors. Meta-analysis looking at many papers up to that point in time of 2017 relating hair cortisol to chronic stress. If stress is still ongoing at the time of sample collection, in other words, continuing stress, the time when you collect the hair cortisol was elevated compared to controls by an average of 43%. Again, consistent with the idea that adrenal increase in adrenal cortisol manifested in the blood is reflected in hair cortisol. Maybe even better, hair cortisol is elevated in people with hypercortisolemia, which means elevated cortisol in the blood. We call this Cushing syndrome. It can occur from a number of possible reasons. And it's also reduced in people with adrenal insufficiency. So this means that either your pituitary is not making enough ACTH or your adrenal glands are not responsive to ACTH. And this is a very deadly disease, Addison's disease, if it's not treated. So if the cortisol was coming from your hair follicles, then Addison's disease shouldn't necessarily have any effect on that. And here we see, this is hair cortisol levels, individual data points. This is healthy individuals, overweight, obese, and this is the Cushing, and different sources, different reasons for the Cushing. It could be a pituitary tumor, adrenal cancer, adrenal carcinoma, and we see mean much higher. They don't show the Addison's disease here, but actually, yeah, that's right. They don't show the Addison's disease, but that also shows a reduction. So it's tracking. These are, hair cortisol is tracking changes in blood cortisol. So we take all that together. My interpretation, this is my interpretation, is hair cortisol mostly, again, we can't know how much might be coming from local synthesis, but at least mostly originates from the blood. Or we can't rule out the second possibility that this HPA axis in your follicles responds the same way that your adrenal glands do. So that's possible. We can't theoretically rule it out. But either way, to me, if we take all this together, it gives me reasonable confidence that cortisol in your hair is not only sensitive to stress, but it's sensitive to stress because it's mainly tracking changes in your circulating cortisol produced by your adrenal glands. All right, this is a good time to take questions, especially with this big issue of local synthesis. As I say, if you read most of the literature, you won't even see any mention of that. So I think many researchers who get into this field either don't know about that or they're happy to not talk about it, because for people who haven't put together all the information that I just presented to you, it could get sticky. It's like, well, how do you know? Reviewers are going to start rejecting your papers. Grant panels are going to start rejecting your grant proposals. So you need to really have all that information at hand to, at least in my case, I'm just speaking for myself, to feel reasonably well satisfied. Kestis, Dr. Bendinskis, one of our participants, is also in the field, and I don't know how convinced you feel about this, but anyway, if you wish to say anything about it, I'm welcome to hear your thoughts.
Yeah, I would go along with you, Jerry, on the fact that our hair follicles probably respond to all other stimuli like glands, and therefore they follow the same pattern of response. So, and therefore it's mostly, it's a good argument, but it may be irrelevant where exactly cortisol is being made, as long as those cells respond the same way. So in a way, I feel that hair cortisol is a proxy for HPA axis, right? It's not an HPA axis, it's a proxy. So if we interpret it as such, then it's perfectly fine. In regard of how, where cortisol comes from, there has been that paper that really bothered me for a long time until I thought I started thinking about it. It's called the Hair Cortisol, a Parameter for Chronic Stress, Insight from Radiometabolism Study in Guinea Pigs. It's from Germany from 2012 by Kekheis. And they used radioactive label to inject, and they could not find anything in the hair follicles themselves. Right. So it was a very strong paper that made me think about this issue. But by now,
well, there's a monkey paper also that failed to find hardly any elevation in cortisol, in radio-labeled cortisol in hair after a bolus, I think it was a bolus treatment of tritiated cortisol in monkeys. And so, so I'm aware of both of those papers. I think the problem with these radioisotope, we shouldn't get bogged down in this because other people will have questions too, but just to say very quickly, I think one of the limitations of that is I think if you want to really do it right, you've got to really elevate you've got to get a high level of radio-labeled hormone in the blood for a long period of time to really test it accurately, to test it properly. And if you can't do that, which is impossible either for money or for danger of having that much radioactivity floating around, you really can't do it. So I think that's the problem with it. But anyway, let's open it up to other questions. Thank you for your thoughts.
Hi, I had a question about DHEA. I noticed a lot of what you were talking about applied to hair cortisol, and I was just wondering if the same kinds of like synthesis process would apply to DHEA in hair, or if there were any other like specific considerations if we wanted to look at DHEA in hair versus looking at
Right.
cortisol in hair.
Yeah, I'm not aware of any not aware of any studies looking at DHEA production in hair. I suspect that the hair follicles probably have the biochemical ability to synthesize it, the machinery, the enzymatic machinery to make it. So, but I don't think anybody has actually demonstrated it or quantified it in the follicles. It is present in the hair. So we would have the same issues, DHEA in the hair. If some of it's being made in the follicles, we don't know how much, but it hasn't been really looked at in the same way that cortisol has in
Okay.
follicles.
Thank you.
Yeah. Yep. Any other questions?
Jerry, could you maybe go
Yeah.
over covariables that everybody needs to know about when collecting data and collecting hair cortisol at the same hair samples at the same time? Because those
Yeah.
are very important. And also the questionnaire that you have for the hair collection, that's extremely important.
Yes. So those are, yeah. Unfortunately, I neglected to make a slide with these issues of covariates, but since you raised the question, I can talk on it a little bit. So I already mentioned that there are differences, for example, in hair growth rates, and therefore also, it turns out, also differences in average hair cortisol based on race. So particularly with respect to African American versus American Caucasians, where on average, African American, have about a 20%, at least the data that I've seen that I believe, about a 20% slower growth rate, and but higher average hair cortisol. Could be related to growth rate, but it could also obviously be related to other things that co-vary with race, other factors. So you want to know about that. Age. There's a paper that I recommend. I think it's cited later on in the presentation from a Swiss group, Tina Binz in Zurich, who has been doing a lot of hair cortisol and nail cortisol. And so she's one of the groups that has published a proposed reference range. That is, people often ask, how do you know if somebody's hair cortisol is high or low or medium or whatever? And this gets into the issue of reference ranges. So in medicine, we have reference ranges in relation to health risk. So if you're at a certain age and your blood pressure is above a certain cutoff that the doctor looks at based on, hopefully based on research literature on mortality and stroke and whatever, that's when they'll tell you your blood pressure is too high, you need to reduce salt or take blood pressure medication, so on and so forth. So the reference ranges for medical things, including hormones too, but mostly blood hormones. So if they want to test you whether you have Cushing's syndrome, one of the things they'll do is they'll take a blood sample, maybe they'll do a urine sample, and there'll be a cutoff. If it's above a certain level, then that's suspicious that you might have Cushing's syndrome. So what about hair? The problem is that hair cortisol is affected by how you process the samples and then your measurement method at the end. So if they're doing like your, if they do a blood draw and they want to look at your blood cells, what's your content of red blood cells? Are you anemic? What's your white blood cells? Are they elevated? Do you possibly have leukemia? And so on and so forth. So it goes into a machine. Machines are calibrated. They have certain standards that the machines have to meet to be used for diagnosis. So that means that you can have a set of values that should be pretty much replicated from machine to machine for something like blood cell counts. We don't have that for cortisol. Again, how you process the sample. Do you grind it up? Do you wash it? Pretty much everybody washes the samples. I'll get into the methods when we come back from break. Pretty much everybody washes hair, but still occasionally people don't. You grind up the hair. How do you extract the cortisol? And then importantly, how do you measure it? I already showed two different ELISAs can give somewhat different absolute values. LC mass spec may give you still a different value. Anyway, so I think, so with the caveats, with all those caveats in mind, if somebody asks me about a reference range, since I haven't published one and really haven't tried to put together one, I tend to refer them to the BINS paper, which I'll cite later on. But anyway, the reason I got into mentioning BINS is because she also, the paper lists some differences related to age. So they looked at toddlers, then they looked at children to young adolescents, and then they had more or less adults. They didn't really look at elderly. But what they showed was in their cohort, which was Swiss, but also, I guess, some samples, Australian samples, I'm not sure why, but Swiss and Australian, the toddlers had higher levels than adults on average. Then children to beyond toddler age, toddler being, I think, about 3 years, they defined. And then children up to, including early adolescents, I think were lower, a little bit lower than adult, and then adult. So there are age-related changes going on during that period of time. Then it's fairly stable over over many years in adults. And then possibly, I think, an elevation when you get into late aging. There's an elevation in blood cortisol, so it wouldn't surprise me that you'd see that in hair. So ethnicity, age, sex. There are sex differences. I say sex here, meaning biological sex. And many studies with small to moderate sample sizes, modest sample sizes, don't find a sex difference. So you don't see it in every paper where it's looked at. But the meta-analysis that was done by Stalder et al., where they analyzed many, many studies, thousands of participants, on average, males had a higher cortisol level than females. So sex is important. And then use of medications is very important. So when somebody's collaborating with us, we send them two things. We send them a recommended method of collecting and storing hair, and we send them a questionnaire to give to their participants, which ask about hair washing frequency, which in many studies has no effect, by the way, even though you would think it might. In many studies has no effect. But hair washing frequency, but importantly, medications. There are many medications that will interfere, and I'm not just talking about things like, you could get effects of taking psychotropic meds, like antidepressants or antipsychotic drugs. It's not well studied what those effects could be, but still, there could be effects of those medications. Recreational drugs, of course, could also have effects. Many recreational drugs stimulate cortisol release through the HPA axis. But also medications that actually contain steroids. So some medications actually contain cortisol. It doesn't usually say cortisol on the label. It will typically say hydrocortisone. Hydrocortisone is the same as cortisol, just a different name. Hydrocortisone is present in many topical creams and ointments. So if you have a rash on your skin or an insect bite or eczema, if you're suffering from eczema, you may use hydrocortisone cream. That gets into your blood a little bit, not a lot, but some. It's on your hands, even if you wash your hands. And now you maybe are putting your hands through your hair, gets into your hair. Obviously, it gets into your nails if you're looking at fingernails. So hydrocortisone medications are, if anything, going to elevate hair cortisol inaccurately. If you're using a different kind of medication, like prednisone, if you're taking prednisone pills for immune suppression, if you're taking steroid sprays, nasal sprays, like I do for asthma, those contain synthetic glucocorticoids that will have a mild suppressant effect on your adrenal output. So those can interfere for a different reason. So our questionnaire actually lists many, many medications to help people remember or be aware of what they might be taking, even if they don't know offhand that it contains a steroid.
Do you use you probably have to get a good health history as well,
Yes.
right?
Yeah, well, we don't. Again, if whoever we're working with, yes. And you want to exclude, any number of health conditions, any serious diseases, endocrine diseases, obviously, diabetes, and diseases of other endocrine diseases that may, even if they're not specifically the adrenal, could affect adrenal function. Yes. So getting overall health information, that's part of the questionnaires, certainly.
And not familial. They don't need anything like that.
No.
Yeah. I'm thinking about the doctor.
Just of the participants themselves. If the only exception would be if we're looking at parents and infants, and we're looking at infant hair or infant nails, we'd want to use if the caregivers, parents or other caregivers, are using especially topical meds that could get onto the infant.
And diet and smoking issues as well.
Smoking, yeah, smokes. Well, sure. Again, nicotine is a stimulant for the HPA axis. So if you can't exclude, you either could exclude smokers, or if you can't exclude them, then you might want to control for it as a variable, confounding variable. So sometimes you just can't exclude everything. Really depends on the nature of the sample you're working with. If you can't exclude all the things that might cause problems, then you're stuck with trying to statistically control for them.
Right. Yeah, I was wondering about
that. Doesn't always work well. But.
Right. I was wondering about the steroids. So if you can, exclude those if you can, even like the hand,
yes.
or the nasal or the eye or whatever. Okay.
Yeah. Yeah. I would say the worst offenders would be either topical medications which are usually over the counter, Cordaid, those kinds of things. I use that myself actually for skin irritation, or systemic steroids, obviously. The use of inhalers has been shown, if you have a good enough sample size, it's been shown to have a suppressant effect, a modest suppressant effect, and to thereby possibly confound hair cortisol measurements. So ideally you would exclude it. If you have a lot of people who are using them and you really can't exclude that many people, then you would enter it as a variable in your regression analysis.
One other question, the hair dye. A lot of women.
Yes. Good one. That's a good one. Thanks for bringing that up.
Yeah.
Forgot about mentioning that one. Yes, we also ask people about hair treatments. So this is the literature on this is actually, once again, it's kind of scattered. Some studies show an effect of hair dyes. Other studies don't. We definitely tell people to ask. We can see it on a sample. The hair dye will be soluble either initially in the isopropanol wash, that's the washes we do of the hair, or if not, then the methanol extract to pull out the cortisol will pull it out. And so we'll see color in the extract. So we make a note of it and we report it back if the collaborator wasn't already aware of it, if the person had not self-identified, or if the person who collected the hair had not noticed it, we'll report back that we've identified that this is, but it's not as if you see hair dye there and you see some totally wacky cortisol value. You don't. So if there are effects, they're probably relatively subtle. I think that probably what's been shown in the literature, to my knowledge, the worst offender would be bleaching. Bleaching is very harsh on your hair. And bleaching would be likely, if anything is going to have an effect, bleaching might be the biggest one. Hair straighteners, I don't think there's been as much research on looking at that. So it's been mostly hair dyes, coloring, or bleach.
Any
Other questions?
nutraceuticals?
Yeah.
Any nutraceuticals that alter cortisol levels?
I suppose it's possible, but off the top of my head, I don't know.
Well, the reason why I ask is because some of our Tourette's patients sometimes consume nutraceuticals that have, either to increase their manlyhood or to combat adrenal fatigue, and it can make their tics worse. And so I'm thinking that maybe, is there like a specific group that we should be asking?
Can you be more specific about the substances?
So like there's what is it? P40, whatever. There's some of these testosterone supplements that are, that are available in the supplement. Would those would it be valuable to assess those kinds of
if you have subjects who are using that kind of stuff, it's going to affect their physiology. It's possible that it could affect their adrenal function. So it's, you can always, so the questionnaire we sent, first of all, people have their own questionnaires for medical histories and other things. So they'll incorporate our questions into what they already have. And you can always add stuff, that you think might be relevant for your population. So there's no harm in doing that, as long as you're not getting to some questionnaire that's so onerous that you start getting people dropping out of your study because they don't want to be bothered as much by having to fill out these forms.
Given that, then you should probably exclude for use of steroids, even if you're doing saliva samples. Is that right?
Yes, it's going to be the same, right? Sure. Anything else?
You had mentioned that.
All right, well, yeah.
Oh, sorry. You mentioned that cortisol was different during pregnancy. Can you speak to what direction that was in?
Yeah, well, what happens is, so hair cortisol is tracking pregnancy-related changes in circulating cortisol. So there have been studies, course, it's kind of handy in a way, since we often look at 3 centimeters representing 3 months. It's kind of handy that pregnancy has got trimesters. Each one reaches roughly 3 months. So there have been studies in which hair samples from the same group of pregnant women were obtained at the end of the first trimester, second trimester, and then at the end of pregnancy, representing and then 3 centimeter samples taken from each to assess the amount of hair cortisol and the amount of adrenal activity in each trimester. And what you see is if you were looking at plasma cortisol or salivary cortisol, you would see not big changes over the first 2 trimesters, but the 3rd trimester, it definitely ramps up. And so if you're looking at hair cortisol, you would expect to see a jump in hair cortisol concentrations, which, as I say, is expected from the increase in circulating cortisol during the 3rd trimester. Yeah. So that's the biggest thing to look for. Anything else?
So
Okay, great bunch. Sorry?
Is there any guideline? There is no literature that I know of, but based on your lab experience, is there any age, cutoff at which we have to start worrying about the older adults having higher cortisol as compared to?
No, because we really haven't encountered any projects that we've worked on that worked on that sampled a quite elderly population. So
Okay.
I couldn't give you advice on that.
All right, thank you.
Yeah, I'm sorry. All right, well, we better move on because there's still a lot of material, and we're going to get into the nitty-gritty of how we do hair cortisol. So those of you who might have an interest in it, or nails, can see the various steps involved, can see why we and Kestis charge money for this. It's very labor and supplies intensive. So let's go on. All right, so first we're going to give a few slides on the methodology, at least what we do in our lab. So first is cutting the hair to length if necessary. You know, if you've gotten a hair strands that, if you want 3 centimeters and the hair is longer than 3 centimeters, then we have to measure and cut, again, from the scalp end, or what we call the root end. Then we weigh the hair to the nearest 1/10 of a milligram. So we have an analytical balance, a couple of them actually in the lab for accurately measuring the hair. We need to know the weight of the sample that we're going to be working with. And we do this in a grinding tube. These are, we grind samples. We started this with our monkey hair, and we found that grinding made a difference, gave us better extraction, better recovery of cortisol that was in the hair. Not all labs do this, but we do. So this is, these are tubes that are specially hardened. And so the little balls are not in there yet. These are for the grinding process, but this is the tube. So then we wash the hair. You can actually see some human hair samples in these tubes. We wash the hair twice with isopropanol. So this is a mild solvent. The idea is to, since there's a good reason to believe that the outside of the hair is coated with cortisol that has come from sweat or sebum, probably mostly from sweat. We want to remove that cortisol while as little as possible removing cortisol that was in the interior of the hair shaft. So we found in our initial studies that 2 washes, each wash is 3 minutes long, with isopropanol seems to do the trick for that. And so we put the tubes on a rotator here, we rotate for 3 minutes, and we pour it off. We do that twice. So then we dry the washed hair at least overnight in a fume hood. That's what we see here, is tubes that are just being dried. So the isopropanol just evaporates. Then the next thing is the grinding. We use chrome steel beads. We put 5 beads in. They're small beads, as you can see. And we use a bead mill. There are several different versions of this apparatus from different vendors. So basically, whatever the apparatus is, it takes the tube and the tube is moved back and forth rapidly. So the beads are flying back and forth inside the tube, repeatedly hitting the hair strands and eventually pulverizing the hair. So depending on what apparatus you're using and what kind of beads you're using, it may take differing amounts of time to get a decent grind. For us, with this apparatus and with the beads we're using and with the tubes we're using, it takes typically 3 runs of 2 minutes each to grind the hair reasonably well into a pretty fine powder. Then we want to extract the cortisol. So we add 1.5 ml of methanol, which in contrast to isopropanol, methanol is a much stronger solvent for steroid hormones. And unlike isopropanol, methanol causes the hair to swell. So we're using isopropanol in part because it does not cause hair swelling. Again, with the idea of maximizing removal of external cortisol while minimizing internal cortisol, which we think might be more liable for extraction if the hair swells. So here we want the hair to swell, so we add methanol, and we extract for 18 to 24 hours on a rotator, so the tubes are just going round and round. We spin the tubes in a centrifuge, and we take off. There's no way you can get a whole 1.5 mils back. So we spin the tubes, which pellets down the hair powder and the beads, and then we pipette off 1 ml of the methanol, and we transfer that to a clean tube. Then we have to get rid of the methanol, so we evaporate it in a vacuum evaporator. That's shown here. You can do it in other ways too. Some people just use a stream of nitrogen gas, but this is more rapid and efficient. And then we add assay buffer to reconstitute the extract. So now the cortisol is tiny amount of cortisol, and it is tiny, that we've extracted from the hair, is down near the bottom of the tube, on the inside of the tube, on the wall of the tube. We need to get it back into solution. So we redissolve it. And then when we redissolve, in our hands, when we redissolve these extracts, we get some particles, which we found interfere with our eventual assay. So many people have not had this problem, and so good for them. But for us, because we have these fine particles, we have to get rid of them. And so we filter those particles using a centrifugal filter. So this is a little tube. We pipette in the cortisol that's back in solution, but with those little particles that are interfering, and we centrifuge this, and the particles are retained on the filter. This is a cellulose acetate filter, and the liquid containing the cortisol goes through, and now we're ready to go. So the assay that we use, as I mentioned this morning, is from Arbor Assays in Arbor, Michigan. It's the one we use for human hair. It's highly sensitive. So it's good when we have samples that are relatively small or that have really, really low cortisol content. And I'm not going to go through the whole assay here, but suffice to say that there are a number of steps in the assay, including mixing a plate. We actually have a new and better plate mixer than the one I show here. Plate washing, this is an automated plate washer. And then the colored product is measured, it's produced in 96 well microplates. I think I have a, yes. This is what it looks like here. This is a 96 well, it's 8x12 matrix. And the way the Arbor Assays ELISA goes, and this is true for the majority of ELISAs for cortisol or other hormones, is that the amount of the hormone content in each well is inversely related to the amount of color that's produced. So the most color, as you can see, the ultimate color that you're measuring is yellow here. More yellow means less cortisol. Okay, it has to do with the chemistry. We don't have time to get into it, but it's what we call a competitive assay. Competitive assays give you this inverse relationship. And here's a standard curve generated from one of our assays. This is the log. So we have standards with known amounts of cortisol. Here's the highest standard. You can see the least amount of color. This is the lowest standard. This is a blank that has essentially no color. That's just for non-specific binding, we call it. And so this is the log of the cortisol standard. These are standards in duplicate relating to the optical density at 450 nanometers, which is the wavelength. There's a laser wavelength that this yellow dye absorbs at. So, and then your unknowns, all of these would be unknowns. Your unknowns are interpolated from the standard curve. So if an unknown's over here on this, this amount of absorbance, the computer calculates where that fits on the standard curve and spits out a cortisol value. Then we take that cortisol value and we use that to determine, we use that with the, along with the amount of hair or nail that we analyzed. That's the last step here, to determine the final value. And the units of measure, I should mention here, units of measure are picograms per milligram of hair. Picograms. A picogram, for those of you who are not used to that unit of measure, is 1 to the minus 12th grams. So we're really talking about very, very small amounts that you're able to reasonably accurately measure using these kinds of techniques, using these ELISAs. You can get even higher sensitivity using the LC tandem mass spec method. So I mentioned earlier that one of the issues why it's hard to develop a reference range that everybody could use is that the levels do depend on methodology. So hair washing, yes or no, say most people do wash. Choice of solvent, some people choose to wash in other things in addition to or besides isopropanol. Whether the hair is ground, minced, one alternative is cut the hair into little pieces, or even nothing, just work with intact hair, and then the immunoassay. So I want to mention for anybody who might be wanting to get into this, either yourself or with a collaborator, do not, under any circumstances, use the ALPCO kit for hair cortisol. Papers that we haven't never tried this ourselves, but there are several papers in the literature in which that kit was used, and compared to any of these others or compared to LC tandem mass spec, this kit for some reason with hair cortisol gives just really high values, much higher than any other. So I was even contacted by some folks, I think in Taiwan, who were trying to set up through hair cortisol, and they hadn't asked me, they asked me about all the variables, but they didn't ask me about the kit. And they said, oh, we're getting these really high values. We don't understand why. They were using the ALPCO kit. So we recommend staying away from that one. Okay, so here's the reference range that I mentioned, so we can just reiterate the issues here. And this is the Binns paper that I alluded to, 2018. They're quite a good lab, and they do use the LC tandem mass spec. So it's good to keep that in mind. The values that we get on average aren't, reasonably within the same range that they get, but also, these are not, if you have a US population, which I assume most of you will be working with, these are Swiss and Australians, and who knows whether environment in different countries, whether diet, any of a whole host of things that differ between countries might affect hair cortisol. We don't know. It hasn't been systematically looked at, as far as I know. here is the reference range that they reported in that paper. So I mentioned that they have toddlers, they have late childhood and adolescents, and then they have adults. So they decided, and it's not clear to me why they decided, it wasn't clear in reading the paper, either I missed it or they didn't explain it, why they decided for the low to be bottom 25th percentile. But they did. And then the middle is the bulk of individuals. And then the high is top 10th percentile. But you see the difference. You see what I said before. Toddlers higher than adults on average, adolescents less. And this shows up in the reference ranges. At some point we have so much data, at some point we might try to put together a reference range. The problem is that we don't usually know after we analyze a bunch of samples what the demographics were of the people who donated those samples. And so therefore it would be just kind of a big mishmash. So that's the reason that's held us back. So hair cortisol, I think, has a tremendous range of applications. One obvious one that many of us are interested in is current life stress. Another is history, which I think was brought up in some previous questions earlier today. History of prior stress or trauma, either in childhood or later in life, but before now. Mood disorders, drug addiction, these are all areas where research has been done. Other illnesses, whether endocrine or other illnesses, HPA system development, just looking at normative changes in HPA function. In utero exposure to cortisol, this is one that's very near and dear to my heart. I've been interested in developmental questions really since I was a graduate student a million years ago, but hair or nail cortisol has potential, which I'll get to in a little bit. Wildlife conservation, our polar bear study was directed towards that. Animal welfare, I mentioned that we've done pigs, was actually a study that's been published, 2 papers in collaboration with an Israeli group that was looking to improve husbandry of breeding pigs and piglets. And we found some very nice data that I hope are being paid attention to by pig breeders. Archival samples. Cortisol is amazingly stable in hair. Doesn't mean that it might not change over very, very long time periods, but even at room temperature, it's amazingly stable. We received in the polar bear study, unbeknownst to us, they sent us some polar bear hair that was taken from museum samples. There was some pelts in this museum in Denmark. It was a Danish research group that we were working with. And they had clipped some hair. And we actually found higher levels of cortisol in those old samples, over 100 years old, than in modern day polar bears. And it's interesting to speculate about why that might be, but it shows how stable it was. Cortisol has also been measured in human hair from mummies excavated in Peru that were estimated to be a couple thousand years old. And cortisol was still measurable in those samples a couple thousand years ago. So amazingly stable, useful for old samples, even at room temperature. So I'm going to give you a few examples, some from my lab, some from other research groups. This is a study we did, published a few years ago with collaborators at UMass Boston. And as you can see, they the UMass Boston folks sampled 180 young adults from the UMass Boston community. These were more or less young adults. and about half male and half female, and almost 2/3 were minority. And participants reported self-reported amounts of lifetime discrimination as well as recent discrimination. We didn't specify racial discrimination, but this was obviously one of the major sources of discrimination. And this is we took the data from lifetime discrimination scores and made tertiles. So this is the bottom third, the lowest levels, the medium levels of lifetime discrimination, and then the highest levels. And you can see the systematic relationship. When you get to the high levels of perceived lifetime discrimination, there was a significant elevation in hair cortisol. Interestingly, there was no change, no difference in hair cortisol when they just looked at recent discrimination. So this is looking at more of a long-term effect on, in this case, increasing HPA activity. And then there's some other issues that were interesting, some other findings from the study. So this is an example from that domain. This is from, I think a Dutch group, I believe, was looking at hair cortisol in shift workers compared to day workers. And you can see shift workers who, have constantly changing when their work hours are, so they don't have a stable day-night cycle over periods of time. And you can see that they had two different age ranges, but the biggest effect was actually in the less than 40-year-old, but also all the data together were significant. So what you can see here is that some of these are earlier studies. Some of these earlier studies were relatively simple in comparing groups that you think might be more chronically stressed versus other groups, control comparison groups that you hypothesize would be less stressed. A lot of the more recent research is using much more complex designs where you really have to analyze your data using multivariable statistics and determining from those kinds of analyses what are the variables that impact significantly hair cortisol levels. But here's another earlier study, a very nice study from Kirschbaum's group when Tobias Stalder was still working with Clemens. And here they were looking at caregivers, elderly caregivers of patients with dementia, typically Alzheimer's disease. And you can see that the caregivers had significantly higher hair cortisol than the controls. And moreover, in those individuals, there was a significant correlation between hair cortisol and caregiving burden, as assessed subjectively, in a scale of that, and also depressive symptoms using the Beck Depression Inventory. Okay, so if you can bear with me a little longer, we've got about 10 more slides, but some really important stuff. So I hope you guys are hanging in there. This is something that I alluded to a couple of times earlier, and I think it's really an important area of the field. And I didn't run across it immediately when it was published, but I recently ran across a study, 2019 study, that I think is quite interesting, addressing this issue. So, so we've already said that many studies have run subjective stress measures, most often the perceived stress scale, that 10 measure scale that goes back a month, because it's so easy, readily available. It's been well validated as a good measure of stress over the past 30 days or so. But the results have been highly variable in terms of whether PSS scores correlate with hair cord. So the idea of correlation is this. It's not just, well, is your hair cord up, but your PSS, in a group. A group has high cord, but no high PSS, or a group has high PSS and no high hair cord. Oftentimes you get elevations in both, but they don't correlate. which means that the extent of the stress effect on the subjective measure for each individual is not the same. It's not the same extent of an effect as the effect of that stress on the biological measure, on cortisol. That's how to think about that. So when we're using the PSS, the PSS is 1 month, as we've said, but cortisol is typically, even in the studies where they're trying to correlate them, 3 centimeters, and therefore 3 months. You could say, well, obviously the problem is because they're looking at different time periods. One's looking at 1 month, the other's looking at 3 months. So if they don't correlate, that explains it. But it may be more than just that. So as I say in this last bullet, can the question be addressed more systematically? And that's where this recent study, this is Weckwesser, I'll give you the reference again, the citation again, published in 2019. And fortunately, I ran across it because I think it's really valuable. Participants were 37 young German couples. So there's 74 total, 37 men, 37 women. They were couples, so they, you know, some may have been married, others were just in a relationship. And they were studied over 12 weeks. So they were studied over basically 3 months. And the investigators really went out of their way to really see, what's the best way to try to relate subjective stress to hair cortisol. So they actually gave their subjects 3 different subjective stress measures. They did the Hassel scale. Remember the Hassels and uplifts scale, which is set up to do a day's worth of Hassels and uplifts. So they gave it once a week, and they focused on the Hassel scores as a measure of stress. They gave the PSS. PSS goes back a month, so they gave it 3 times. They gave it at the end of every 4 weeks, okay? So they got past this issue of only give it once at the end of the study and you've got 3 centimeters of hair. So they don't correspond. They gave it 3 times. And then they gave this Trier chronic stress measure once at the end of the study. And they collected hair sample week 13. So they're going back over the 12 weeks, basically, of the study when they had these subjective stress measures. There was the big paper, a lot of results. So obviously, I'm not going to try to give it all to you. This is what I think was the most informative graph for our current, and also some text for our current discussion. So one thing that was interesting, I'm not sure what it means, but the PSS, just look here first. The PSS and the tics were correlated with each other more strongly than with the Hassel scale. Not sure why, but that's part of the data. The only subjective stress measure that by itself that correlated significantly with hair cortisol was the weekly Hassel scale. And it wasn't a great correlation, okay, but it was significant. Furthermore, if they look at, so of course you can square this and get a variance estimate. So what this means is that 84% of the variance of hair cortisol, if we think about this relationship, is not explained by subjective stress. I'm going to come back to that in a second. But here is, they have a lot of graphs, but here's the one that I thought was most interesting. The Hassel scale, remember, what was done weekly, but then they also had the PSS monthly, so going back 4 weeks, and they somehow combined these. It wasn't clear to me how they combined the various subjective stress measures, but my understanding of the graph was that's what they did. And here is the correlation week by week with the hair cortisol. So these are correlation coefficients here. These are individual data points week by week. This is the confidence interval here. I added, this is my addition to the graph. This is where the correlation peaks. And so the correlation in this study peaked about 4 to 5 weeks when it was measured 4 to 5 weeks before hair collection. So all I'm saying is, at least based on this, okay, there may be obviously maybe other considerations. Based on this, you might think about collecting, if you have 3 centimeters of hair and you have some reason to relate the subjective stress over that 3-month period to your hair cortisol collected here, you might want to do your subjective stress assessment here rather than here. Okay. I don't know. But anyway, that's what they actually found empirically. So to me, I think, yeah, okay. So to me, how do we interpret this? The fact that even doing all of this, the best correlation they could get, significant, but still modest at best, 0.27. What does it mean that we don't have a better correlation? So they talk about, psychological traits. They talk about recall bias. All these things are legitimate factors. I would add, being a neuroscientist by training and by orientation, I would add that the brain mechanisms, we know a lot about the brain mechanisms that are involved in stress responding, that is subjective stress, like anxiety, fear, things, going through the amygdala in a large degree, and the mechanisms by which stressors converge on the PVN to affect CRH, et cetera. Those systems overlap for sure, but they're not identical. There's differences in the neural mechanism, the neural pathways that give rise to subjective stress versus cortisol release or any other biological marker. So this is further evidence, I think, further rationale why you want to do both. That you're not getting a complete picture if you're either doing subjective stress by itself or if you're doing biological stress measures by themselves. You really need both because I think they're giving you complementary information. When something is only modestly correlated, it means that there's a lot of information that you're not getting from each one alone. So that's my spiel about, and when we're done in a few minutes, I'll be glad to take any questions about that issue. So I'd like to turn to a little bit about development and try to go through this quickly because I know it's getting late. As I said, I'm really interested in developmental questions, developmental studies. And many researchers have been interested in this, especially the impact of prenatal stress, that is stress of the mother on the developing fetus and how that impacts the offspring at birth, as well as the offspring's development over time after that. So the majority of this research uses looking at cortisol in the mother, traditionally saliva. Nowadays, you could also look at hair in the mother. But there's a problem, and that is that the mother's cortisol is only partially responsible for the cortisol that the fetus is exposed to, especially once the fetal adrenal starts becoming active, mainly in the 3rd trimester. So by the time you get into the 3rd trimester, the fetal adrenal is ramping up cortisol production, and it's a major contributor to the fetus's exposure to cortisol. So you're completely missing that. Plus the placenta metabolizes a lot of the mother's cortisol as it passes through the placenta. There's a placental enzyme that is responsible for this. And so if women differ in their placental cortisol metabolism, again, the amount of cortisol the mother produces is not totally reflected in how much the fetus sees because of placental metabolism. So we believe, we are arguing with only minimal data so far, but we believe that there is a rationale for assessing newborn hair or nails. Many babies are not born with any hair, but you can get nails. In fact, probably a few nails collections by the parents are still reflecting prenatal nail growth. Maybe a better measure of fetal cortisol exposure, mainly during the third trimester and maybe a little earlier, because hair and nails begin to grow about mid-gestation. And so you could get some exposure prior to the beginning of the third trimester. So Melinda and I wrote a review paper on this topic, and it's in your reference list. If you're interested in this, I highly recommend that you read this and see what we have to say about it. So interestingly, everybody agrees with this, that hair cortisol levels in newborns are much higher than those in their mothers. We first saw this in monkeys, in newborn monkeys compared to their mothers, but it's also been shown now in newborn human infants. And one theory is that this is due to cortisol in amniotic fluid. There is cortisol in amniotic fluid. The baby is bathed in amniotic fluid, and some of that may well be getting into the hair over time and reflected in the newborn's hair cortisol. Give you an example. So Sonia is one of our participants, and she, I'm sure, would be happy to see this slide here. So this was from a study from Sonia and the group, Kim Noble's group at Columbia, that was published a couple of years ago, actually 1 year ago. And I give a little bit of information about this. So the participants were 60 mothers and their infants, 6 to 12 months of age, from New York City, a variety of outcome measures. And the important ones for us here are the hair cortisol, the mother's PSS scores, perceived stress, and EEG that Sonia collected, and that Kim does a lot of work with EEG as well as neuroimaging. So what I'm showing here is kind of a simplified version of the data for purposes of presentation. So as infants within this age range, as they mature, the EEG transitions from having high power in the theta rhythm to increased power in the alpha rhythm. And we see that here. So here is, this is theta, relative theta power, different parts of the scalp, the EEG measurements. Of course, you do Fourier analyses to determine the amount of power, the amount of rhythm in a given wavelength band. And here's the alpha power. So the difference here is that when we did a median split of the mother's hair cortisol, so these are the mothers in the lower half of their hair cortisol, mothers in the higher half, and comparing on average their infants. And we see that the mothers in the high hair cortisol group, their infants show a delayed transition from theta to alpha, indicating a delay in their brain maturation of this sort. So this is just one example of the power, the usefulness of hair cortisol in developmental studies. Nails. So very quickly, nails are becoming, it's still relatively early in the field, sort of like where we were just a couple of years into the hair cortisol field. Nails are a potential alternative. And reasons for this: sometimes you can't get a hair sample. Newborn infants, many don't have hair. Men can pose a problem. Either they shave their head, or they're bald, or their hair is very short, so you can't get 2 or 3 centimeters if that's what you want. So the point is that cortisol can be measured in fingernails and toenails. We use very similar, not quite identical, but very similar processes in processing nail samples compared to hair samples. Oh, I meant to show here, we like to use fingernails for a couple different reasons, but if someone is contemplating using toenails, it's important to realize that there's a different growth rate. So you can't compare them. So if some of your subjects give you fingernails and others give you toenails, you've got a problem. That'll be one or the other. This just shows the growth of a nail. So the part that we're mostly aware of and that we actually clip over here is called the nail plate. This is this brittle material that has a lot of keratin, which gives it the brittleness. Then we have the nail bed, which is tissue just underneath the nail plate. And the germinal matrix, these are dividing cells, which give rise to the nail plate. So this would be sort of comparable in a way to the follicle cells in the skin that make the hair shaft. So nails, so hair grows pretty much linearly. It grows out of your skin one direction. Problem with nails is they grow in 2 directions, outward this way, but also they thicken. And that complicates the determination of when the cortisol that you're measuring, when you clip nails over here, when was it deposited? We think that cortisol is getting in at the very least from the germinal matrix cells here, as well as some incorporation perhaps from the nail bed. Some evidence for this, but it's not conclusive. So our best estimate, other investigators differ a little bit. Our best estimate is, based on our interpretation, is that cortisol in the nail sample that you've clipped out here has accumulated over maybe about 3.5 months, very roughly speaking, prior to the clipping out here. Some argue longer than that, maybe 4 or 5 months. So there's different estimates in the literature based on different data. Features of fingernail cortisol. Like hair cortisol, there's evidence for intra-individual stability over time. Here's some unpublished data from our lab from one of my honor students last year. So she had fingernail clippings at 2 months taken from the same participants at 3 months, 3-month intervals. And here's the matrix, and you can see a pretty decent correlation, 0.5, between the log of the second value versus the log of the first value. Unfortunately, a later sampling using mostly the same participants, not everybody was giving their nails at each sampling time. A later sampling didn't find it. So we're kind of stuck. This study is actually ongoing, and so we'll have more data at some point. By next year, when we give this presentation again, I should have more data on that. Some studies show a significant correlation between hair and nail, but the results are variable. So some studies don't show that. And importantly, there's a lot less information on whether chronic stress elevates nail cortisol compared to what we know about hair cortisol. We've done a little bit of, several groups have done a little bit of research on this. And I'll just end before the summary slide. I'll just end with this. So this was a study on refugees living in an asylum center in Serbia. The refugees were from the Middle East, 111. And it was decided that they were more likely to be willing to provide nail samples compared to hair samples. The outcome measures included the PSS, fingernail cortisol, and reports of traumatic experiences, either at their home country before they emigrated or during their journey. The PSS score, which is shown here, and the nail score, which didn't quite make statistical significance, but it was a strong trend in the same direction, were higher in the refugees who had experienced trauma during the journey. And these included physical hardships, violence, and so forth. So the traumatic experiences at home did not significantly show up in these measures, but in their emigration prior to arriving in Serbia, showed up as higher perceived stress and a strong tendency toward higher nail cortisol. So this paper as well is in your folder. So finally, some conclusions. I'm not going to read all this. You can read it. It's pretty much summarizing what we've said. Hair, we've talked about hair treatments can be problematic. Nails have problems too. Nail treatments, especially nail polish remover, which contains acetone, which is a solvent for steroids. People, especially most typically women who are wearing artificial nails, they don't want to take them off. So, so it's not immune from all problems, just a different set of problems compared to hair. So anyway, thank you very much for hanging in there. And I hope that you found this session informative and useful in your own thinking about stress research. So especially incorporating HPA axis measures. So questions. So we have, the schedule says break and then a Q&A. Well, given the time, we're just going to go right into the Q&A, and then we'll be finished for the day. So feel free to ask about anything that I've talked about, but most likely I figure if you have any questions, it'll be about the chronic stress and stuff I talked about there. Everybody just too tired? That's okay, if it is the case. I'll give you a minute to collect your thoughts, see if there is anything you'd like to ask me about. Of course, you have my email address, so you're certainly welcome to contact me. I'm happy to try to answer any questions you might have, to elaborate on any of the topics that we covered. The papers that you have access to should be very, very useful in helping you delve into this field. And of course, there's a huge literature beyond, especially hair cortisol, beyond what I've given you and what I've discussed here. So I cherry picked, we all cherry picked to make the points we want to make. But I think I've been, tried as much as possible to be fair. And certainly we've discussed some of the limitations, experimentally, limitations of interpretation. We should always keep in the back of our mind. Confounding factors, questions were brought up about that, so we had a chance to talk a little bit about those. So if anyone actually, occasionally participants in this workshop decide that they actually want to incorporate this sort of thing in their research, and but they don't want to do it themselves. So if you're interested in doing that and possibly collaborating, of course, you're welcome to contact me. Anyway, last call for questions.